Pharmacy Universitas Andalas as your master of ceremony for the conference. First, let us say thanks to the Almighty God who has been giving us guidance, happiness, health, and mercy so we can join and participate in this event today. And let us send our salawat to our Prophet Muhammad sallallahu alaihi wasallam. It is an honor for me to introduce and welcome all honorable guests in this event. First, Honorable Director of Universitas Andalas, Eva Yonedi SE MMPM Akuntan PhD. Second, Honorable the First Deputy Dean of the Faculty of Pharmacy, Universitas Andalas, Professor Apoteker Yufrialdi, MSc PhD. Honorable Chairman of Committee International Conference on Contemporary Science and Clinical Pharmacy 2024, Dr. Apoteker Dwisari Dilasamola, MPharm. Honorable all the deans of pharmacy faculties and institutions in Indonesia. Honorable, our Excellency Speaker, Insinyur Audi Joinaldi, SPT MSc MMIPM ASEAN Engineer PhD, as the Deputy Governor of West Sumatra, Indonesia. Professor Jung Woon Lee, from College of Pharmacy, Seoul National University, South Korea. Professor Apoteker Armenia PhD, from Faculty of Pharmacy, Universitas Andalas, Indonesia. Professor Muhammad Makmur Bakri PhD, from Faculty of Pharmacy, University Kebangsaan Malaysia, Malaysia. Professor Zahir Uddin Babar, from Department of Pharmacy, University of Qatar, Qatar, and University of Huddersfield, United Kingdom. Professor Pauline Lai, PhD, from Department of Primary Care Medicine, University of Malaya, Malaysia. Dr. Clinfarm Apoteker Dedi Almasdi, from Faculty of Pharmacy, Universitas Andalas, Indonesia. Professor Apoteker Andi Dian Permana, PhD, from Faculty of Pharmacy, Universitas Hasanuddin, Indonesia. And Associate Professor Sukanya Dech Adisai, PhD, from Department of Pharmacognosy and Pharmaceutical Botany, Prince of Songkla University, Thailand. Honorable, our Excellency Invited Speaker, Apoteker Islamuddin Ahmad MSE, from Faculty of Pharmacy, Universitas Mula Warman. Apoteker Dira Hefni MSc PhD, from Faculty of Pharmacy, Universitas Andalas. Apoteker Tasia Amelia MSc PhD, from Sekolah Farmasi Institut Teknologi Bandung. Apoteker Purnawan Pontana Putra MSi, from Faculty of Pharmacy, Universitas Andalas. Dr. Rizky Edmi Edison PhD, from Faculty of Medicine of Universitas Prima Indonesia. Apoteker Penny Indrayuda M. Biotech PhD, from Faculty of Pharmacy, Universitas Muhammadiyah Surakarta. And last but not least, Apoteker Rini Agustin MSc PhD, from Faculty of Pharmacy, Universitas Andalas. And especially for all participants who have been joining us in this special event. Excellencies, distinguished guests, participants, ladies and gentlemen, thank you very much for your participation on the second day of International Conference on Contemporary Science and Clinical Pharmacy, or ICCSCP 2024. Today, we have several agendas to do, including the plenary session 3, followed by parallel session 2, plenary session 4, award presentation, and closing ceremony. Ladies and gentlemen, to begin our activity today, let us say Basmalah. Bismillahirrahmanirrahim. Excellencies, distinguished guests, participants, ladies and gentlemen. We are now beginning our conference and move to the next agenda, the plenary lecture session 3. The plenary lecture session 3 will consist of 2 lectures. The first lecture will be delivered by Professor Pauline Lai, PhD, from Department of Primary Care Medicine, University of Malaya, Malaysia, and followed by the second lecture by Dr. Klinfarm Apotheker Dedi Almasdi from Faculty of Pharmacy, Universitas Andalas, Indonesia. In this session, we would like to extend an invitation to all attendees to post questions by entering them in the chat room. These questions will be addressed during the upcoming Q&A session. For this session of plenary lecture, we will be led by Apotheker Yoneta Srangenge, MSc. Ladies and gentlemen, please allow me to introduce our moderator. Apotheker Yoneta Srangenge, MSc, is an avid lecturer at the Faculty of Pharmacy, Universitas Andalas. She achieved her bachelor degree at Universitas Andalas and continued her master degree at University College London. She has done some research and publication in clinical pharmacy and pharmacology. Her latest publications include evaluation of analgesic use rationality and level of patient knowledge in primary healthcare facility in Padang in 2021, and identification and education of adverse drug reactions and drug-related problems in the elderly community at the Lubuk Begalung Elderly Posyandu Padang in 2023. Without any further ado, I would like to invite Apotheker Yoneta to be on the screen and guide the plenary lecture session 3. To Apotheker Yoneta, the screen is yours.
All right, thank you, Jihan. Sorry for the delay. There is some technical issue in here. All right, Assalamualaikum warahmatullahi wabarakatuh. Alhamdulillahirabbil alamin wassalatu wassalamu ala asrofil anbiya'i wal mursalin wa ala alihi wa sahbihi rasulillahi ajmain. Asyhadu an la ilaha illallah wahdahu la syarikalah wa asyhadu anna Muhammadan abduhu wa rasuluh la nabiyya ba'da amma ba'du. Good morning, everyone. Welcome to session 3 of the International Conference on Contemporary Science and Clinical Pharmacy 2024. And my name is Yoneta Srangenge, and I'm honored to be your moderator in this session. And as Dian already said, we have two distinguished speakers in this morning who will share valuable insight in the area of pharmacy informatics and pharmacovigilance. And each speaker will present for 45 minutes, followed by 15-minute Q&A session. And all of you can submit your question at any time to the chat room, and we will address them during the discussion period later on. And if you like to ask a question directly during the Q&A session, you can raise your hand, and I will call you at the appropriate time. All right, without further ado, let me introduce our first speaker who already joined us in the Zoom in this morning, Professor Dr. Pauline Lei. Good morning, Professor. All right. Professor Lei brings over 20 years of experience as hospital pharmacist before transitioning to academia in 2010. And University Malaya Medical Center, she was also instrumental in advancing the department by implementing a computerized pharmacy system and prescriber order entry, or we call the CPOE system. And since March 2023, she has been a member of the FIP Technology Advisory Committee, where she applies her expertise in implementation science. And her current research focus on harnessing smartphone application to empower patients in self-care. Today, Professor Lei will present on pharmacist informatics in hospital health system, the opportunities and the challenges. And we look forward to exploring her insights into how technology can bring integrated into hospital health system for better patient care. Professor Lei, thank you for joining us today. Can we start the presentation?
Sure. Thank you, Yonita.
All right.
Okay, I'll share screen. Can you see the slideshow?
Yes. Can we make it fuller? All right.
On my screen, it is full. Is it on the slideshow already, or is it on the PowerPoint presentation?
It's already in the slideshow.
Okay. All right, can I start?
All right. Yes, please, Doctor.
Okay, good morning, everyone, and thank you very much for the kind introduction, Yonita, and thank you for the organizers of ICCSCP for inviting me to talk to you all today. So I'm going to be talking to you about the pharmacy informatics in hospital health systems, what the opportunities and challenges are. So although Yonita has actually mentioned a little bit about myself, I just thought I'll let you know that I was a hospital pharmacist for 20 years before I decided to get my PhD and move into academia. So whilst I was a pharmacist in the hospital, which is a tertiary hospital in Malaysia, it has over 1,600 beds then. Now it's almost a 2,000 bed hospital. I was actually the first pharmacist to be the pharmacy information manager, which means that I actually converted a totally manual pharmacy into a computerized pharmacy. Then once we actually did that with a lot of hard work, I then implemented the computer prescriber order entry, also known as e-prescribing or CPOE, throughout my hospital. After which I got my PhD, went over to academia, became a member of the FIP Technology Advisory Committee, and my research expertise now is in implementation science, and I utilize technology like smartphone applications, websites to engage patients to self-care. So in 1999, to err is human, and all of us are human. So we actually, in the United States, it was recognized that medication errors are the largest single cause of medical errors in hospital, accounting for more than 7,000 deaths annually. And so healthcare organizations and healthcare professionals like you and I must implement proven medication safety practices to reduce reliance on our memory, to standardize terminology, to use constraints and forcing functions, protocols, and checklists, and to minimize data handoffs. So in 2001, there was a new committee that actually came up with a new health system for the 21st century. And they recommended that the use of technologies and systems should be utilized to reduce medical errors. And that includes having an EMR, an electronic medical record, so that it can store patient-specific clinical information. And that EMR should be integrated with a computerized prescriber order entry system, which then links to the pharmacy to tell us how to dispense the medicine and to distribute the medicine. And then once it gets to the ward, for the nurses on how to administer the medicines. So this is an example of an EMR and a computerized prescriber order entry by the doctor. And these systems should work together within and across organizational boundaries. So for instance, like one hospital talking to another hospital or one hospital talking to a clinic, share real-time patient information, and provide timely, relevant clinical decision support at the point of care. So in America, after 10 years of effort, the deployment of CPOE and clinical decision support systems in the US was only about 15%. In beds with more than in beds that have more than in hospitals that have more than 200 beds. And adoption of CPOE in ambulatory care was greater than hospitals, where over 25%, 1 in 4 office-based physicians use an e-prescribing system, and nearly 70% use an e-prescribing application within their EMR. How about medication conciliation? Only 10.4 hospitals had an entirely electronic process for medication reconciliation, and most hospitals implemented a pharmacy information system, but only 50% could interoperate with their EMR, while the remaining pharmacy systems were standalone systems, only for use within their pharmacy. Most hospitals adopted medication distributed and storage technologies, about 80%, repackaging automation for unit dose medications. However, integration was low. Only 20% had all systems in place. For electronic medical records, 8 to 12% of US hospitals have a basic EMR. Only 2% have a system that meets federal government's meaningful useful criteria. Larger hospitals, urban hospitals, and teaching hospitals were more likely to have EMRs. So what are the barriers and challenges to actually having an information system within the hospital? Firstly, obviously, it's going to be financial because it costs a lot of money. It also depends on your workforce, the strategy, the cultural component, the structural component, technical component, and the privacy and security issues. And I'll go into each one of this in the next few slides. So the cost of implementing health information technology varies. It varies on the size and complexity of the operation, the replacement costs for the hardware, the computer hardware, etc., and the extent to which the organization chooses to perform its work electronically. In 2005, the cost of implementing a CPOE system in the US was about $63,000 a bed, and this is in US dollars. So the financial liabilities of hospital information technology is more than the fixed cost of hardware and software because it has to include maintenance, hospital staff for training and temporary reductions in productivity when the training occurs, the inability of healthcare providers to capture immediate financial returns, and the costs associated with state and federal privacy and security requirements. How about workforce challenges? There is insufficient supply of health information technology professionals who have education and training that intersects healthcare, computer science, and informatics. We have a lot of healthcare professionals who are very good at what we do, but do we know very much about information technology? Not really until we actually get into doing it, like myself. Okay. Similarly, we have a lot of people who are very good in computer science, but do they understand very much of what we do in healthcare, and so on and so forth? How about strategic challenges? There is an overall lack of vision for what an ideal technology-enabled medication use process might look like. Healthcare organizations work more as a group of independent departments rather than a tightly connected process for healthcare delivery. Rather like this. Hospitals are challenged with delivering quick technical solutions in response to a serious patient safety or regulatory requirement. We fail to consider the impact of their decisions on future connectivity efforts. Healthcare professionals have also traditionally practiced within their functional silos and to support specific roles and responsibilities. So when we implement a new computerized system, or we try to computerize our workflow with the latest information technology, it is a major cultural event. It requires committed leaders and an environment for innovation, willingness to forego perfection in pursuing modest incremental improvements. Structural challenges here I am referring to is with regards to the organization's data that are secluded in different departmental systems. So health information exchange is the anytime, anywhere access to clinical information across traditional business boundaries. And this is actually essential for advancing hospital information technology initiatives. For technical challenges, existing hospital information technology rarely takes advantage of the human-computer interaction principles. It results in poor application design and greater pushback from the end users. Current hospital information technology architectures lack the plug-and-play capabilities. It is developed in isolation and are not randomly harmonized with coexisting systems. And of course, we are all concerned about privacy and security, where this is a really important step for users, healthcare professionals like you and me, to use the system to protect the patient's identity and anonymity. We need to follow a country or a state's law to protect patients' information, and different countries have different requirements. So what do we have to do to overcome these health information technology challenges? We need to recognize that these health information technology has a major impact on pharmacy practice, and accept that this current and emerging technologies would supplant roles that were traditionally performed by pharmacists. We should resist waiting for the perfect solution to become available before pursuing any health information technology applications. And we should continue to seek these solutions to have incremental gains to ensure that these gains are aligned with our institution and ideal HIT strategic objectives. And we should also articulate an ideal vision and strategy for an IT-enabled medication use process. We as pharmacists should influence regulatory groups, HIT vendors, health system leaders to pursue sound methods for achieving optimal HIT approaches to medication management systems. We should work collaboratively with community and health system pharmacy leaders to achieve a higher level of medication system connectivity and to integrate technical and semantic medication standards to support the system. Pharmacy should build and strengthen relationships with internal and external stakeholders that influence HIT development, pursue leadership positions within the HIT industry, and advocate for new professional roles for pharmacists in informatics and clinical analytics. So what is health informatics? Health informatics bridges the healthcare with information technology to improve clinical care, ensure patient safety, and increase the efficiency and effectiveness of organizational processes. Pharmacy informatics is a subset of health informatics, and it requires both our clinical expertise and knowledge about information technology to improve medication management processes and drug administration safety. Pharmacy informatics are people like you and me with a solid background in clinical drug distribution systems. So what are the core competencies of a pharmacy informaticist? In order to enable growth, these are the 5 areas of competency, which I'll go through one by one. The first one is data, information, and knowledge management. For data, information, and knowledge in this particular section, I would like to highlight to you the ones that I've highlighted in bold, a lot of words, but interoperability, data governance, and control terminology of certain words that we use, standardized terminology. So firstly, management of the drug database. Developing a standardized drug and order set nomenclature in the drug database is absolutely essential because we need to provide a consistent description of a medication and avoids confusion within and between systems. So this is an example of a label from my hospital, and we label all our medications generically. And of course, that is our label for our hospital. And we capitalize certain words within the drug name to emphasize that this is chlorpheniramine. And of course, we actually emphasize telan 1 tablet sekali sehari pada waktu malam. You can tell that our labels have both Malay and English, yeah? Okay, so generic names should be used. These are some of the guidelines that we follow in our hospital. An approval process for the use of medication abbreviation and truncating rules that prioritize the display of a drug salt, formulation, extended release modifier or strength. And if let's just say there were any new drug additions to be added into the system, it needs to undergo an evaluation process where it is counter-checked to prevent selection errors by end users, because published literature has shown that when a new drug is added into the system, if an error occurs at that point of time, the error can be compounded. So this is an example of another label in our hospital. We had bisoprolol fumarate, and that is capitalized as well to actually display the drug's salt. The other keyword that was bold, that was highlighted just now, was interoperability. So the system should be able to talk with other systems. So for example, the EMR, the electronic medical records, need to talk to the pharmacy information system. And with that, the CPOE, which is the computerized prescriber order entry, it would be ideal to have a clinical decision support system. So imagine the patient coming to see the doctor. They have the electronic medical records. The doctor has a talk with the patient and finds out what's wrong, etc., etc. Has a look at what the patient is currently taking on the pharmacy information system. The doctor then prescribes the medication, and at the point of prescribing, there is a system to sort of like tell the doctor, okay, what you prescribed is too much. There's a drug interaction between this drug and the patient's current drug, etc. While prescribing, the doctor can also have a look at the laboratory information system to have a look at whether the patient's creatine clearance is good or not good, or whether any dose adjustments need to be made. And finally, prescribes the medication. And if the patient's in the hospital, a nurse would then administer the medication to the patient. Moving on to the second competency, information and knowledge delivery. Again, many points, but what I actually wanted to highlight was the clinical decision support. So with the computerized prescriber order entry and clinical decision support, we have this big wheel, which is the CPOE, and we also have the clinical decision support, where information delivery can be provided before decisions are made, or passively as reference information. So a passive reference information in Malaysia, in my hospital, we use the Micromidex and the British National Formulary. This might not be what you use, but that's what we use. And if let's just say a hospital can, if they have insufficient budget, they can start off with a passive reference information, better than nothing, but at least it's an online reference source that doctors can look up at the point of prescribing. But however, if one can have a clinical decision support system, it would be obviously better. So a clinical decision support system aids the clinician during the prescribing. It's an event-driven alerts, which means that when the prescriber prescribes two medications that interact, a pop-up message will occur. There's a forcing function, which means that let's just say the minimum and the maximum range of the drug is set within the computer system. So if the prescriber accidentally types in 200 instead of 20, an extra zero, then it will force the prescriber to prescribe maximum 20, okay? There are care plans that you can actually set within the system, which means that plan A will go to plan B, will go to plan C. Evidence-based order sets. We can also have documentation templates and patient data summaries. Now, so this is how the CDS works. There's a trigger in ordering medicine, input data, creatine levels, current medications, and then this intervention is the CDS gives option in terms of drug interactions, allergy verification, and critical laboratory values. And the action step is to change those. However, it is very, very important for us as pharmacists to monitor and evaluate the effectiveness of these clinical decision support systems based on user feedback and adjustment required, because alert fatigue and data overload can develop. So let me just share with you a bit of my experience. Initially, when we actually got our system with this clinical decision support, we had a lot of alerts, and doctors initially, it was like if the doctor prescribed amlodipine, which is an antihypertensive, then they prescribed telmisartan, which is and they prescribed, say, bisoprolol. Because both were antihypertensive medications, the system actually alerted the doctor, both are antihypertensive medicines, but it is very common for a patient to be on one, two, or even three antihypertensive medicines. So those alerts didn't make sense and they became very annoying. So the doctors actually provided their feedback to us, and we removed some of those that we felt that were not clinically relevant. Okay, let's go on to the next competency, which is practice analytics. Okay, I would like to actually point you to this word called business intelligence. So practice analytics is actually business intelligence. It's a process that uses technology and database creation to study clinical and fiscal processes to improve decision making. So examples was documentation of clinical pharmacy KPIs within the EMR. When the EMR allowed clinical pharmacists to do this, they could document their initial patient assessment with follow-up notes, select which KPIs have been completed throughout a patient's admission, and extract and audit their services to improve patient care and safety. Another example is the enterprise data warehouse, where by having an information system, you're capturing a lot of data. So we already now have a large repository of business, operational, and clinical data for research, quality improvement initiatives, and predictive analytics. So the data generated needs to be reviewed by a pharmacy informaticist to provide background understanding to ensure queries are accurate and used in the correct context. And in my institution, we actually utilize a lot of this data for our research and to see trends in medication use and management as well. moving on to the next core competency, which is applied clinical informatics. Again, these are the words that I would like to highlight: to analyze problems and to produce solutions. So when we apply the clinical informatics, it improves our clinical practice with the usability, efficiency, and safety of systems by applying user experiences research and theoretical informatics principles. An example is automatic dispensing units collaborating with nursing to develop key principles for system configuration and decisions surrounding emergency overrides. The other example is the use of auto substitution when a medication goes out of stock, which in our hospital, it happens very often, or therapeutic interchanges. And if any case, if we have drug shortages, what to do. The last core competency is management of change. To lead local and external organizations. Pharmacists need to participate in impact analysis and change initiatives while also providing oversight and leadership concerning medication management systems. For example, this IV bag here, revision of IV bag labels to include diluent volume plus overfill to actually fill up all these tubes here because these are dead space. Labeling change was then evaluated by a committee and a clear communication plan was developed of how batch labeling are done, the worksheets, and for the IV pumps. So what are the future directions? Pharmacy or health informatics courses should be offered in pharmacy undergraduate courses to enable a shift to a more fully supported pharmacy practice. Now, I don't know about you, but I never had this in my undergraduate years because laptops were a rarity in, I don't think it even existed when I was undergraduate student. But however, undergraduate curricula now has to actually look into this because this is more and more urgent. Big data can be used to improve clinical decision-making and pharmacy research. AI, artificial intelligence, is the thing that's coming up now, and you'll be hearing more and more about it. They are the future of healthcare, and pharmacy informatics are essential to development and adoption of AI. That includes drug design, formulary selection, choice of drug therapy, treatment predictions and results, healthcare data processing in terms of diagnostic tests, wearable devices, and natural language processing, potential drug interactions and adverse events, and adherence monitoring. And we need an increased representation of pharmacy informatics as leaders. This is just an example of an organizational chart that I got from literature. This should actually, this shows an example of what an organizational chart can look like. So we have a chief pharmacy officer and a director of pharmacy and a chief pharmacy officer for information. So this is just as important as being the chief pharmacist. And the chief pharmacy informatics actually reports to both. And you'll have the ambulatory care, medication management, outpatient, reporting, and oncology. Then there's a pharmacy information resident. I must confess to you that even with my hospital that has been computerized for over now 15 years, we do not have the luxury of this organizational system where so many people are actually involved in, where so many pharmacists are actually pharmacy informaticists. Okay. And with that, I'd like to move to my conclusion that practice of pharmacy informatics is in a state of rapid growth. The diverse and evolving field leaders that use technology at multiple levels of practice needs to be from department projects to national collaboratives. And pharmacy informaticists, people like you and me, are in great position to collaborate with other healthcare providers to optimize information management, improve workflow, and reduce medication errors. And when we support and develop the pharmacy informaticist role, the profession of hospital pharmacy can optimize innovations to medication-related processes so that pharmacists can continue to improve patient care and outcomes. And with that, that's the end of my lecture. Thank you.
Thank you, Professor Lei, for that enlightening presentation. Now we will move to the Q&A session. Please feel free to type your question in the Q&A chat room, or you can raise your hand if you would like to ask directly to Professor Lei. And I will start with the few questions to get us started. So, Professor, before you mentioned about that your experience in clinical expertise before you transitioning to the academia, I really like wondering why you take that kind of decision.
Because it was not the pharmacy information bit that I was bored. I was bored with the normal pharmacy work. For me, I like pharmacy. I have always liked pharmacy, but it was no more mentally challenging. So I decided I was talking to my friends and colleagues about it, and they said, I think you're bored. You need something more to make your brain active. So they asked, why don't you do your master's? Because I have a bachelor's, I don't have a master's. So when I was doing my master's by research, which I could do part-time, my master's project turned out to be really, really good, that my supervisor asked me, why don't you convert it to PhD? And I said, oh, can we do that? She said, yes. So when I did that, I actually converted my master's project to a PhD, got my PhD, and then I went, okay, so what do I do with my PhD? Then my colleague in the university said, why don't you come over to academia and help us teach, because we are so short of PhD people with qualification. And that's how I moved. So everything was an accident, Yonita. I didn't aspire or I didn't dream to become an academician, an academic person. But when I did it, I found that I loved it. So I'm an accidental academic person.
Wow, that's so interesting.
Just my story.
Yeah, because so like before you also mentioned that actually I just wondering like there are so many stages for us to develop the CPOE system, right? And to be it like fully implemented in the hospital setting. So for the initial movement that we can do, because like in Indonesia, especially in Sumatra, we already start about the computerized system, but it's not about the prescription, direct entry. So what do you think that the step that we can take to to get to where you are now?
I understand your question because we were there as well. Okay. It is very, very important that your boss, for us, it was the director of the hospital that supports your decision as pharmacist, because pharmacist is actually only one department within the hospital, isn't it? So you need to start with having to say that you want to computerize pharmacy. That is the most important thing, because pharmacy in any hospital has a lot of budget, because we buy the medicine for the whole hospital. Okay, so we have millions of ringgit a year to actually handle. And without a computer system, it becomes very messy. So we actually told the director of the hospital that we want to computerize our pharmacy. Then when we actually were looking for vendors to actually supply the computer system to us, they said that with our computer system, it comes with a e-prescribing system and a clinical decision support system. So then we went back to our director and said, look, you gave us this amount of money to buy the pharmacy information system. Can we top it up a little bit and we can get the e-prescribing system as well as the clinical decision support system? Then our director, I must say, was very forward thinking. He knew that information technology was the way to go. And he is a doctor himself. He is an endocrinologist. He actually said, look, okay, we'll give you an extra budget. So you roll out pharmacy information system first, then we roll out the other thing. So it was step by step. You have to do things step by step. But you need the money. And who's going to give you the money? Your boss is going to give you the money, in the sense that your boss has to ask our ministry for money. You get what I mean? So it's one step at a time, and it took years, Unita. It didn't just happen overnight. And for us to actually have the pharmacy information system, I think I had a lot of white hair then, because I was leading the team and we were staying till really late at night, to 8 p.m., 9 p.m. Then the next day we had to come in at 7 a.m. So we had to work really long hours. But once it actually took off and we had to train all our staff, then after that, once that got stable, we went to the e-prescribing system, then we went to the clinical decision support system, and it took years. But we have matured to today to a system that is stable. And whenever there is electricity interruption, like no electricity, so we can't use our computer system, all the doctors cry because they're so used to having a computer system and all the decision support.
Yeah, exactly. I'm not thinking about the electricity turn off before. Like, oh, it's gonna be so hectic, right?
Yes.
All right, thank you, Professor. Now we have a question for in the chat room. Before we let one of our participant to ask directly to you. So it's from Irene Puspa Dewi. She asked about how about the management of narcotic drugs? Is that the specific handling for this drug? And because in Indonesia we have no monthly report for this drug.
Okay, thank you for the question, Irene. Narcotic drugs are treated exactly the same way as any drug in the computer system, but in addition, according to our laws for the Dangerous Drug of Addiction Act, we have to have a manual book. So we record it twice, once in the system and also in the manual book where it's signed by 2 pharmacists, etc. Now, you have to comply to the Indonesian drug laws. So whatever the drug laws that exist in Indonesia, you have to do that. But if you have to have a monthly report for the drug, if you have it in the system, you can just press a button and the computer, if you configure the report to what you want, the computer system can just print a monthly report for you anytime. So it even makes your life simpler in the management of narcotic drugs.
Yeah, but somehow we already like we also like afraid in the other hand, because like is it gonna be like make us so reliant to the like to the what's called technology?
Yes, it
Yeah.
does. Let me ask you a question. If let's just say if you don't have your phone now, do you feel as if something's missing, your mobile phone? Yes, right?
Yeah, yeah.
So we are all now dependent on technology. You know, my son cannot live without his mobile phone. All of us can't live without our mobile phone, our laptop, our iPad. So yes, we are dependent. We are no longer independent. So we just use technology to the best of ability to help us work in a more efficient manner.
There's something that we cannot ignore as well, right?
Cannot escape anymore.
Yes, so we have Dr. Dwi Sari here with us. She wants to ask you a question. Please, Doctor.
Okay, thank you, Ms. Yonita. Good morning, Prof. Pauline.
Good morning.
Thank you for your insightful presentation, Prof. I have a question regarding 2 questions. Number 1, what specific opportunities have emerged for pharmacy informatics with the rise of electronic medical records (EMR) in multi-hospital systems? And the second, what are some of the major challenges faced by pharmacy information is in ensuring compatibility and interoperability across different hospital systems? Thank you, Prof.
Okay, so sorry, Unita. Can you repeat that question, the first one, because I couldn't hear very clearly. What was the challenges? Is it?
Okay. What specific opportunities have emerged for pharmacy informatics with the rise of electronic medical records in multi-hospital systems?
Okay, we'll do one question at a time. So what are the challenges of pharmacy informatics is with EMR, is it?
Okay, yeah.
Okay. EMR is not so much with pharmacy informatics, it is actually from the doctor's end. So it is to actually collect information with regards to the patient, their patient details, the diagnosis, the treatment, and the care plan. Now, how pharmacy comes in with the EMR is that we have clinical pharmacists that go on the wards. So they need access to these care plans to know what the patient has been diagnosed of, what the medicines that they are on, and then if there are any drug interactions, and to supply the medicines. So that is where in my lecture just now, I was actually saying that it can be used to document the clinical pharmacist's documentation of their medication review for the patient. Okay, so for a pharmacist informatist role, that is probably the role of how they can do it then. But essentially, an EMR record is actually for the doctors to use, primarily the doctors. Does that make sense? Yeah.
Okay, thank you, Prof.
Okay, and the second question was?
What are some of the major challenges faced by pharmacy informatics in ensuring compatibility across different hospital systems?
Okay. The compatibility issue, I think, is the data exchange between different organizations. Now, I must confess to you, in Malaysia, between organizations, we are very poor. In fact, almost nil, nothing at all, because each institution still operates in silos, and we don't trust our information going to another hospital, etc. So in order for that to happen, it needs to occur at government level. So in Australia, when I went to the FIP meeting, not this year, but last year in Brisbane, the Australian government is a top-down directive to actually say, look, hospitals need to talk to each other, and hospitals and GP clinics need to talk to each other. And perhaps we need to have a centralized e-prescribing system so that the patient can go to their nearest community pharmacy and get their medicines dispensed from there. Now, doesn't that sound fantastic? So which means that if I go and see a doctor in this big hospital, but I live quite far away, I can go back to the pharmacy near my house and collect my medicine, because everything talks to everything. You know, that is so fantastic. Even in Malaysia, that hasn't occurred. And I reckon that this will take years to come, because everybody is very possessive. Oh, this is my data. You can't have my data. Oh, what happens if you do not take care of my data? Then it's no more confidential, you know? So it's a long journey for all of us, including for us in Malaysia. But maybe Australia is actually doing a better job, and we should look towards Australia on how they do it.
So actually, is it already clear, Dr. Dwi Sari?
Yeah, yeah.
All right.
Okay, thank you,
Thank you, Doctor.
Prof.
All right.
So actually in Indonesia, we already tried the online referral system between hospital. But the problem is when a patient referred to another hospital, the other hospital is so taking much time to accept the request. And also like it's already like a full book and everything like that. And then it end up by the patient is not treated. So it's like we already tried, but we like in so many areas. So hopefully we can improving the system in Indonesia as well.
Yeah, I'm sure. So there must be some sort of an alert system. So let's just say if one hospital talks to the other hospital must receive the alert immediately and act on it. Because if they don't, then what's the point of actually electronically alerting the other person? Isn't it? You want to send things electronically so that it's instant. If not, then you might as well just give the patient the letter, and you tell the patient, go to the other hospital, you'll get your service faster.
Yeah. So, Professor, before you also, we read in your CV that you mentioned about smartphone application in engage patient in self-care, right? So can you share a little bit about specific example or the result for this implementation?
Okay, I have currently two projects ongoing with my PhD students. One of them is for HIV infected individuals. So my student has developed a smart app on the phone, and that teaches that. So there are several components. The first one, so imagine if an individual has just been told that they have been infected with the HIV virus. So they are going through a lot of turmoil, denial, whatnot, then they don't have information. So the first part is education. And we do education module in small packages. We do a video, we do a game, there's a bit of gamification. And then once the patient feels as if they've got enough knowledge, there's another module now that allows them to key in their blood works, their blood results. So particularly for HIV infected individuals, they need the CD4 count, whether it goes up or low. So that's the other module. The next module is about the medications, what medications they're on, what side effects, how they look like. So they have another module. And we also have a adherence score. So HIV infected individuals, they take a lot of antiretroviral medications. They can be on 4, 5. So we actually get them to remind them, this one is once a day, this is twice a day. When you take it, then you just tap on your mobile phone, then it gives you an adherence score. Then lastly, there is another module for NGOs, non-government support organizations, that they can ask for help, or peer group that they can reach out to. No peer group, because it's anonymity. So it's NGO support groups. So with this kind of module, something on the phone, patients see their own results, they know what medicines they're on, they're educated. So it encourages them to care for themselves more, not just to depend upon the doctor to tell them what to do. You get what I mean?
Right.
Yeah.
Thank you, Professor. It's so interesting. But what do you think about the low income setting places? Like, how do you see the role pharmacy informatics evolving over like several years from now?
Oh, I think it's just going to grow, because like how we were talking about that, we are now all utilizing technology. I don't see us using less technology. I see us using more. And in fact, in the FIP Technology Advisory Group, we are all talking about AI now. So AI is going to come in more and more, and we should use AI to make our lives simpler, not to complicate it. But it means that certain roles within pharmacy, the traditional roles, are going to be replaced by AI. But we shouldn't, as pharmacists, we shouldn't be afraid of this change. We should embrace it, but we should think about other ways where, if we free up our time to do mundane roles, then we can utilize our time to do different things that requires thinking power. Yeah. So I think that the pharmacist informaticist role is going to grow. Absolutely.
So actually, this is my last question for you. So like, because like for you, you start from the practitioner to the academia, right? So in here, I think most of the participant and also the Faculty of Pharmacy, we are from academia who want to contribute to this pharmacy informatics. So like, what do you think to contribute to this development of pharmacy informatics?
Do you have a good working relationship with anyone in the hospital side, in the practice side?
So actually, we have a educational hospital in Andalas University.
Okay. So then you need to talk to the people at the hospital side to ask them what they want to do. Then you as academicians can then help in to actually computerize the work, etc. But I can tell you that when I actually computerized the pharmacy, I actually did it from the hospital point of view. We had no academics teaching us, So this is what we call a champion, a pharmacy informatics champion. You need to actually get one person, if you can even get a group, and definitely you need to get a buy-in from the chief pharmacist that they want to do something. Then there is a working collaboration between academia and the hospital side. Okay, but for me, in my particular experience, it was the hospital side that wanted it, because we were doing the day-to-day operations for the hospital. We were seeing the patients every day. We were buying the medications. We were dealing with patient complaints, drug medication errors, etc. Academia didn't have any of that. They were just teaching. Yeah. So because we actually saw all those problems, we wanted to change. So when we did the change, we did it without academia. Okay, but on your end, it's different. It's the other way around. So you actually have to then sell to them the idea that maybe it's time for a change.
Yeah.
Yeah.
All right. At least we tried, right, Professor?
Yes.
All right. Thank you, Professor. So this is the end of our session. Actually, I have so many questions to ask, but yeah, we limited in the duration of the conference, right? So thank you, Professor, for the comprehensive answer and for sharing your expertise with us today.
You're
And
most
on the behalf
welcome.
of organizing committee, it is our pleasure to present you with a certificate of appreciation for your valuable contribution to ICCSCP 2024. Please present the certificate. All right, thank you so much, Professor.
Thank you. Bye-bye.
Hopefully we can have you in the next conference. So
No problems. And if you have any questions later, you can always email me. Yeah.
all right, thank you, Professor.
Okay. Bye-bye.
All right. All right, everyone. We now move on to our second keynote speakers, Dr. Apotheker Dedi Almasdi, MSc, PhD, who already with us in this room. Dr. Dedi is an associate professor in clinical pharmacy at Andalas University and currently serves as the chairman of regional board of the Indonesian Pharmacists Association of West Sumatra, and with a PhD in clinical pharmacy from University Sains Malaysia. Dr. Dedi research interest spans diverse area. We can see in the slide that including antibiotic use, adverse drug reaction, and pharmacist roles in smoking cessation. And today, Dr. Dedi will present on the role of pharmacists in pharmacovigilance, and his insight will cover how pharmacists play a crucial role in monitoring and in improving drug safety. Dr. Dedi, we are honored to have you with us today. How are you?
Okay, fine.
All right. Thank you, Doctor, for joining us today. Can we start the presentation, Doctor?
Okay. Yeah.
All right, please. The screen is yours.
Okay. Okay. Can you see my my PowerPoint?
Yes. Yeah. Okay. That's clear now.
Okay. Thank you, moderator. The next introduction for I am Perian. In this session, the committee give me topic the pharmacist role in pharmacovigilance. Before we talk about the pharmacovigilance, the history, we must know the history of the many aspect drug reaction after drug came to the market. We know in clinical trial we have we have many steps, but every step not sure, not have limitation that data, because the subject of in the research is limitly. So after a drug distribute to customer in the market, many of the drug have the problem there. In the history, we can read, we can see many of drug is recall from the market. This is the some the drug already recall in the market. In 1960 thalidomide, because produce the pharma for Comelia baby. There's abnormality hands and feet, congenital head problem, eye problem, air malformation, etc. After that, in 1997, Serifestacin recall from the market because rhabdomyolysis. In 1998, terfanidin and 2004 revosoxib. All this medicine recall from the market because we found the ADRs, alpha drug reaction, the from many customer, So in the reference, we see the prevalence of alpha drug reaction. 20% of patient hospital have the ADRs, and 5% of hospital admission because the ADRs. One in 1,000 of death in medical ward caused by ADS. In most case, the ADS caused by iatrogenic injury. And it is specific for someone because the specific of his or him genetic. So talk about the adverse degradation is like the iceberg. We in top we see just a little, but in reality many problem and not see in the above. In in this study we see several ADRs affectation from many some countries, like United States America, kemudian Australia, kemudian United Kingdom, report the many number of ADRs in hospital admission, in hospital admission. So to solve this problem, we must construct the patient safety, in any word we can call the mainstream of safety, patient safety fields. Critical component, the patient safety critical component of quality as defined by WHO. System design, systematic factor that contribute to safety, product safe and drug, device, vaccine, and other biological safety service, inpatient and outpatient medical practice, non-personal service, safe environment of care, facilities, waste management, and environmental concerns. These many area must we concern. We have focus or concern construct patient safety. So no, we we uh, we must we must include we must know the pharmacovigilance. Yeah, pharmacovigilance is come from the word pharmakon, Greek, yeah, poison, drug or medicine. Kemudian vigilance from the Latin word, the main is keep to act. So pharmacovigilance is knowledge and activities to detect, assess, understand, and prevent the adverse drug reaction or other related issue with the machine. Uh, so, so purpose of the pharmacovigilance is to improve patient care and safety, especially in use drug and all treatment intervention. Yeah, to improving public health and safety, especially in drug use, discovers problem related to medicine, medication use, and communicates finding to in a timely manner, for to contribute to assessment and the benefit, harm and efficacy, and risk of drug for prevent adverse effect and to maximize the benefit of the drug. Uh, uh, to encourage encouraging safe, rational, and more effective, including cost-efficient use of medicine. And the last, to improve understanding, educational, and clinical training in pharmacovigilance and effective communication with the community. So, concern of the pharmacovigilance include the drug, not only the drug, but include the biological product, herbal medicine, traditional medicine, dan health supplement and medical device. There is pharmacovigilance activities broadly fall into categories: pre-market and post-market. So we can see for the activity of pharmacovigilance, assess evidence, register, after register, monitor, after monitor, change to product information, safety, and alert. Maybe as mentioned before, maybe recall, yeah, because some ADRs adverse declaration, some adverse declaration, and then enforce compliance. This is cycles of the pharmacovigilance activity, yeah. Pre-market and post-market marketing, after pre-market and post-marketing of the drug. So the scope of pharmacovigilance is want to substandard and counterfeit drugs. So medical errors, report about the lack and drug efficiency, yeah, and the use of drugs with unapproved indication, assessment of the rates due to drug use, drug abuse, and interaction with other drug and food. So main purpose of the activity is to ensure patient safety through identify and evaluate the adverse drug reaction from medicine in the market. So activity of pharmacovigilance includes medicine monitoring, safety medicine monitoring, risk analysis, and assessment of medicine usage, and taking the necessary action. So there the early term is we see there are three terminology in field of pharmacovigilance, but this terminology is interchangeable. advanced direction, area, yeah, the advanced direction just the drug day and layer. This is the all grade area is drug advanced drug reaction and this medication all. So we can see in the diagram interaction from the terminology of adverse drug reaction, adverse drug event, and medical error. So in terms of the adverse drug reaction classification, they are commonly the scholar or the researcher commonly this classification in two types, yeah, that what we call a type A reaction and type B reaction. On the type A, yeah, less predictable because this is pharmacological expanding the pharmacological effect, usually dose depend and incident and morbidity of effect, but low in mortality. The example of TPS is respiratory depression, yeah, with opioid analgesia, yeah, cold with SA inhibitor. So in type B reaction, usually unpredictable, might not be picked up by toxicological screening. Incident and morbidity is low, but mortality is high. Example is penicillin allergy and many type of allergy. Others allergy, yeah, can develop the anaphylaxis shock. Yeah. Other classification is E, B, C, D, E, F. E is augmented, B, bizarre, C, continuous, D, delayed, A, ending use, F, failure of efficacy. Yeah. What lead the TPA predictable and TP non-predictable, like I say before. Yeah. So uh, to we must we must know the factor predisposing to IDRs, like age, memory, yeah, gender and race, frailty and undercurrent disease, abnormality, metabolism and response to drugs, renal and hepatic impairment, polypharmacy, previous history of EDRs, expectation, genetic and allergy, and pharmaceutical factor. Yeah. In Europa, classification the level of risk of the have a risk of the ADRs, yeah. Very common if the risk above 10%, common 1 to 10%, uncommon 0.1 to 1%, rare 0.01 to 0.1%, and very rare, and then 0.015%. So so researcher also grading the severity of alpha degradation, to 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 help we to classification the types and awareness in in service in a hospital or in the service healthcare. The grade minor no therapy, antidote or prolonged observation is required. Moderate, this character is required change in drug therapy, specific treatment or prolonged hospital stay. And severe, potential life-threatening, cause permanent damage or requires intensive medical treatment, and lethal, directly or indirectly contribute to death of the patient. So, uh, 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 in evaluation of ADR, many methods, many approach has used in has used by a practice. Yeah? So in this is this uh, in this in this table, in this slide, we can see some evaluation of the air, like algorithm, causality assessment, yeah, Bayesian methods, expert judgment or global observation, clinical studies, and experimental research. Related ability assessment, some technique has has used analysis without explicit criteria, assessment by consensus, preventability linked to error, standard of care, medication related factor, information technology, categorization of harmful treatment in explicit list, combination of more than one of these approaches. So this study and data sources for the raising awareness and assessing the nature and scale of harm to patient. We can see in this development report, patient safety literature, aspect event review studies, WHO program, then local awareness at country level. We can do awareness of international attention for patient safety, high profile case media attention, legislation and compliance, existing research and data source relevant to the adverse event. In terms of assessing the nature and scale of harm, some method can use, some approach can use. Yeah. Study with record or document review as central component, retrospective record review, confidential inquiries, studies of error-related litigation and complaints data. Yeah. Study with data gathered actively from the healthcare professional as central component, prospective record and review comment with interview, cross-sectional survey, then observational studies, yeah. Also analysis of routinely collected and access data, and also report system, like the external to organization, internal to organization, professional, yeah, and key informant method, interview, yeah, and self-administration questionnaire to professional. This the most popular, yeah, in our practice, yeah, method to assess the ADRs, yeah, what we call the Naronjo aspect direction scale, yeah. This 10 question are the previous conclusive report on this direction? Did the adverse event appear after the suspected drug was administered? Did the expectation improve when the drug was discontinued or specific antagonist was administered? Did the adverse event reappear when the drug was administered? This is in some term, other term is recall, call and recall, recall, yeah. Are the alternative cause that come on their own have causal direction? Did the reaction appear when the placebo was given? Was the drug detected in blood in concentration known as toxic? Was the reaction more severe when the dose was increased or less severe when the dose was decreased? Did the patient have similar reaction to similar drug in many previous exposure? Was the adverse event confirmed by any objective? So all the equation we can get was minus, what minus, 0, plus 1, plus 2, etc. So interpretation we can make interpretation if as definite if the score 9 above, probable, and 5 to 8, possible 1 to 4, and doubtful. And doubtful is 0. Yeah. Other method commonly used to assess of the adherence is WHO causality assessment. Yes, WHO developed a method to assess the causality of the expert degradation. We see we can see the category certain, probably likely, probable or likely, possible, unlikely, conditional on or unclassified, accessible and unclassified, classifiable. My each of the classification have criteria, and we can use flowchart to assess this. Yeah. To access WHO use your flowchart to help we to use the WHO causality assessment. So not only two method as I mentioned before in assess the alpha radiation. Many method we can see in literature to has developed to analysis to classify the drug-related problem. This is in this slide we can see Russell-Oclaf causality assessment method, Hedwig at the air severity assessment scale, Liverpool at the air causality assessment, Smoke Thornton ability tool, Cut lesson classification, Hella scale, etc. Many other methods. But the most popular in our practice is Naronjo algorithm and WHO causality assessment, like I called before. So WHO have the program, we call the WHO program for international drug monitoring schema. In this slide, we can see that the level of center for report of DS, National Pharmacovigilance Centers. In Indonesia, this is one of the function of the BPOM, Badan Pengawasan Obat dan Makanan, and then in the international level is where we have headquartered in Geneva, and then to international center in Uppsala. In reporting, we in the reporting of the end we can see this from the national to WHO, then last the Uppsala. But in analysis to verify if the case conform ADR, as federation is from USALA, WHO, and national pharmacovigilance centers. This is a functional pharmacovigilance system. We can we in this slide we can see the ecosystem for in pharmacovigilance system. We see people include the function and the structure. In function, we get reporting, data collection, causality analysis and risk assessment, and then decision making and appropriate action, and then prevent the outcome is preventive medicine related problem or reduce morbidity and mortality. So in people include in this function is reporters, doctor, pharmacist, nurse, other health care worker, consumer. Evaluator, the evaluator is medical specialist, clinical pharmacologist, pharmacist, and epidemiologist. So in aspect structure, we can see many structures involved in this activity. Manufacturers, reporting, detection, in reporting, manufacturer, hospital, and institution. In data collection, causality analysis, and risk assessment, we have the structure involved, the pharmacovigilance center, drug therapy committee in hospital, safety advisory committee in national levels. So in decision making and appropriate action, we can see the sector involved is regulatory authority, industry, health service, professional group, like ID, Ikatan Dokter Indonesia, IAI, Ikatan Apotek Indonesia, Pharmacist Association of Indonesia, Doctor Association of Indonesia, and media. Other is the NGO, NGO, non-government or organization in Indonesia. We call LSM, like Yayasan Konsumen Indonesia, Yayasan Konsumen YL, Yayasan Lembaga Konsumen Indonesia, like this, Yayasan Lembaga Konsumen Indonesia. So many like I say before, many key players in pharmacovigilance. The government is as a central, Indonesia Badan POM or Kemenkes, Health Ministry or Health Ministry, and then is academician. Academician also involved in this activity, pharmacy industry, yes, health professional, and society. All of these key players must work synergist, must have synergist work, synergist work. So in health services, pharmacovigilance must involve in every activity health service. We can see the pharmaceutical standards, pharmaceutical service standard, pharmaceutical standard in hospital, pharmacies, community health center, including pharmacy service. In Indonesia, we call MESO, monitoring efek samping obat. Not only in pharmaceutical service standard, also adverse effect. Adverse effect are documented in patient record as required by policy. Adverse effect are reported in time frame required by policy. This is parameter related to pharmacovigilance in hospital accreditation. In health center, also in health center accreditation, also in community health center, Puskesmas accreditation. One of the goals of community health center accreditation is to improve quality patient service and safety. So, what is the rule of pharmacist? We ask what the rule of the pharmacist. Many rule, many work can do by the many activity can do by the pharmacist. Analysis of each report ADR, identification of drug and patient at high risk of being involved of others, development and of policy and procedure for ADR's monitoring and reporting program, description of responsibility and interaction of related health professional and risk management in ADR's program, use of ADR program for educational purpose, development, maintain, and evaluation ADR record with the organization, organizational dissemination and use information obtained through the ADR program, and then reporting the schedules or unexpected ADR to national center. This is all. This is a role of pharmacist. So this is a key point for prescriber and pharmacist and the safe use of medicine. Yeah, consider whether drug therapy is really necessary. Always consider whether any new symptoms the patient is experiencing could indicate an ADRs. Be aware at risk patient group. Take care with drug known, produce predictable ADRs. When possible, use of familiar medicine. When possible, use familiar medicine. Check the patient history of idiosyncratic reaction or allergy. Because it's more like I say before, the many did in hospital caused by the idiosyncratic reaction and drug allergy. As if the potential is taking other medicine, including some medication of over the counter medicine, ensure that patients are not exposed to risk prone unnecessary drug use. Ensure the patient are informed about the risk benefit of their medication. Identify patients who may have a compromised ability to take or use medicine. Ensure the patient are counseled appropriately on the correct use of their medication. Try to ensure that there is true concordance between prescriber and patient. Check whether there are any specific monitoring requirement. Be vigilant for suspected ADR. If appropriate, we report them to the appropriate regulatory authority. So pharmacovigilance regulation in Indonesia include the area pharmaceutical industry and health professional. We can see some regulation from Indonesian in Indonesia from Ministry of Health and BPM. We can see for pharmaceutical industry, the two regulation from the Ministry of Health, drug registration and pharmaceutical industry. From the regulation of from the BPOM, the regulation of the head of the BPM, we can see implementation of pharmacovigilance in for pharmacy industry. For health professional, we many regulation for had developed, had the made of by the health minister of health Indonesia. Like pharmaceutical service standard in hospital, pharmaceutical service standard in pharmacy, pharmacies, pharmaceutical service standard in community health center, and then the pharmaceutical service standard in clinic, patient safety in hospital. So in our operation, our experience, in our experience, in less than years, we have done some study related pharmacovigilance, yeah, both at the hospital and the community health center setting. The study have done in wide retrospective and prospective method using medical record or interview. Yeah. In the hospital, the aspect drug reaction study has done to stroke ischemic, congestive heart failure, coronary heart disease, arrhythmia, and cirrhosis patient. We also have identified the medicine which have adverse drug reaction on geriatric and pediatric patient. While in the community health center, we has done to the evaluation the ADRs after COVID-19 vaccine. Ya. We call Indonesia KIPI ya. Kejadian ikutan pasca imunisasi ya. KIPI ya. So this is our research, our study related pharmacovigilance before since 2018, study of gastrointestinal bleeding in the use of antiplatelet and anticoagulant is we use a method retrospective, parameter patient compliance in order and laboratory data. Samples 43 medical record. The result we found the 15 patient has experienced effect of gastrointestinal bleeding. In 2018, the study of aspect drug reaction in ischemic stroke patient at Hospital X Padang. Yeah, we did with method prospective use Anaronjo algorithm. The sample is 32. Yeah, we found the ADR of like anemia, urticaria, nausea, rash, edema, insomnia, dyspnea, and the degree of possible and probable. We also study of aspect drug reaction in pediatric patient in Hospital X Padang. The method we use is interview with the 30 health worker. The result almost all respondents know and have encountered aspect drug reaction. Drug that frequently cause there is antibiotic, analgesic, chemotherapy, and anticonvulsant. So also a study aspect drug reaction of anti-arrhythmic drug at hospital X patient. We use method retrospective, parameter is patient compliant, sin order and laboratory data. The sample of 84 medical record, we found the drug causing ADR is amiodarone, digoxin, verapamil, propranolol, diltiazem. The rest statistical difference based on ADR with the age group and duration of therapy. We also study aspect drug reaction on coronary heart patient at Hospital X Padang. The method we use we use is the retrospective. Parameter is patient compliant, sin order, and laboratory data. The sample of medical record is 53. We found the drug causing ADRs is atorvastatin, simvastatin, aspirin, clopidogrel, nitroglycerin. They were statistical difference based on age group. So study ADR on geriatric patient at internal medicine in patient at Hospital X Padang. We use method interview with 24 health worker and review of medical record. We found the most important, most informant knew know the term of IDR, but they had never encountered it. Yeah. Study of the medical record found that some patient use insulin experience hypoglycemia. Yeah. You also do make expert agreement on drug that frequency cause ADR in pediatric patient. We use DLP methods. Yeah. So from the result is research, there are 10 drug frequency cause ADR in pediatric like amoxicillin, captopril, ibuprofen, cotrimoxazole, luminal, metronidazole, paracetamol, prednisolone, zinc, sodium and sodium bicarbonate. The study of study of hepatotoxic drug in patient with liver cirrhosis at Hospital X Padang. We use a retrospective method, use 62 patient medical record. We found the 47.4 patient medicine have been potential induced hepatotoxic, including paracetamol, ceftriaxone, and levotaxine. At the study in geriatric patient, Hospital X Sorolangun, we interview with 13 health workers. We found all informant know about alpha degradation and have also encountered it. Although rarely, yeah. Drug that cause ADR is antibiotic. Some di Mengkulu, the research, the same research we has done in Bengkulu, Hospital X Bengkulu, and Hospital X Bukit Tinggi. So in terms of community health center setting, we did some study related to pharmacovigilance activity. Yeah? This is post-immunization aspect that event in COVID-19 vaccination at community health center A, B, C, D, E, in Padang. The method we use is structured in preview by telephone. Yeah. We found the pain, sore, set injection set, fever, headache, malaise, drop, and disease. After patient after they use a vaccine COVID-19. Yes. And then also also in health the community health center setting, we also did a study aspect degradation using captopril at the community health center. Yeah? We use method of prospective, use Naranjo algorithm. The sample is 44 patients. We found 40 patients experience IDF. Total case is 26. The degree of probable and possible. I think, thank you. This is the last my presentation. This is my team, yeah, student, and collaborators. This is some from the lecture in Faculty of Pharmacy and hospital and M. Jamil Hospital. Arisman, Yeli, Oktavasari, Dwisari, Dila Samola, Ansen Nasif, Armenia, Fitri Ramani, Najitul Tirtia, Dita Permatasari, Dian Ajita, dan and Rasmi Osman. Student, many student we include in this study. This is my reference, detection of new audit detection, a practical guide, patient safety, rapid assessment method, WHO guideline for safety monitoring herbal medicine, handbook of international pharmacy practice, drug interaction, expectoration, practice standard of ASP, pharmacopoeial, definition guide, and clinical pharmacy service. Okay, thank you, moderator. The time I give back to you.
All right. Thank you so much, Dr. Dedi, for the comprehensive presentation. And we will now open for the floor for the Q&A session. And all participants, please submit the question in the Q&A chat room or raise your hand to ask directly. And I will start with a few questions to begin our question. So, Dr. Dedi, you already said about the importance of the pharmacovigilance in pharmacy practice, right? So, I'm wondering, is already like a current initiative or like a pilot programs, especially in West Sumatra, which is focusing on improving the pharmacovigilance practices?
This is some problem in pilot project because this is related to policy of institution and their founding. We just in this we just we found there some evidence many ADR for the next step. As you mentioned, we must make a pilot project. But the problem is I think the fish problem is finance. The fish problem is the policy of institutional policy from the hospital. The set of we make the pilot project. And then and then is related the finance of the project, because the in Indonesia some problem is they finance they how to call the laporan keuangan. I think this is more problem in laporan keuangan. We must make the report not through actual report, not through actual report. We some the report that we make not send the the problem is system. You can our research project that the dana they found came in July or August, July, but the report must finish by September. What do we do? Okay, I think this is the problem.
There are so many factors.
It's here by political decision, by in university or the national in health in Kementerian Pendidikan. Okay, thank you. I think this is my response, my answer.
All right, Doctor. Hopefully, from your comprehensive research, we can propose like a pilot study in the future. So
yes, I hope. But if the system not change, I think we still in hypocrite system. Hypocrite system. Yeah, I see. I hypocrite system. It's still I think is the true in the field, in the lapangan, in the field. Okay, thank you.
All right, Doctor. So there are some question in the chat room already. I will read one of them is from Professor Ahmadi. So yes. Professor Ahmadi asked, sometimes ADRs are related to genomic factor, right? Yes. And even associated with the off-label use. And so Professor Ahmadi want to hear your opinion on how we should address these issues.
Yes, many like I said before, the most fatal of the ADR is idiosyncratic and allergic. Yes, this type caused by the genetically. So allergy, I think we like yesterday from Zahir mentioned, we must conduct the personalized not Zahir and Bakri, personalized medicine. We must conduct, we must do the personalized medicine. We not enough and only to refer to literature, paracetamol, 200 milligram, 40 times, or maximum to 2,000 gram for every day. Not enough. That must we give therapy specific by everyone. We call the personal medicine or tailored medicine. Yeah. So it's but the problem is the cost is high. Yeah, and Indonesia is of Indonesia I think so far ya masih jauh ya because masih jauh apa bahasa Inggrisnya. So
Still far away.
Yeah, for the food, only for the food is a problem. Yeah, but for many patients in Indonesia.
All right, doctor.
Yeah.
So even though it's like still far away, but it's not impossible, right, doctor? Inshallah.
Yes, I think yes.
So like there is another question is from Dr. Najmiatul Fitria.
Yeah.
Doctor, she asked, is the Naranjo algorithm still relevant for today's since it was published in 1981?
Recently published. Yeah. We depend on the journal. Depend the journal we submit. Yeah, depend journal we submit. If the journal is if we need to publish we article, we see the journal, the policy. What is policy? If the journal policy not we not agree with the policy of the one journal, we can choose the other journal. I think the simple. So maybe this the in aspect is in aspect academic. I think in the academic this my answer. But in politics, so we many problems. The reputable journal not very reputable. Not only tidak pasti semua jurnal reputable itu juga memberikan apa. We can hear before Inul Daratisa publish one article in Scopus journal related to paddy. You know, this is the technique use some people, some researcher not agree with the no system. So, to describe how system is how the our system. I in some discussion I say in economic we call the economic balloon economic is the big but not contribute not in a big but we not way. And if kena duri ya meledak gitu. So in academic, I in our academic atmosphere like this. Yeah. So we we busy with international publication, but our community poor. Our community is not help. Our community is not have have the good service in hospital, in clinic, in puskesmas. I think like this.
Not yet ya, dokter ya.
Yeah,
Insya
not
Allah
yet.
will be.
Not yet.
All right. So we would like to please Dr. Dwi Sari to ask the question directly.
Yeah, the last question
Please,
ya,
dokter.
Miss Kepo.
All right.
Okay, thank you, Dr. Dedi. I'm
Thank you, Mr.
appreciate your
Chairman, Mrs. Chairman.
presentation, enlightening presentation. I have two questions. My questions are number one, pharmacovigilance. We know that pharmacovigilance is essential in monitoring and preventing adverse drug reactions (ADR) to ensure patient safety. This involves detecting, assessing, understanding, and mitigating risks associated with medication use. What are the main objectives of pharmacovigilance and why it's important for patient safety? And then, pharmacists play a crucial role in pharmacovigilance by monitoring drug safety, conducting risk analysis, and taking action to address medication risk. How do pharmacists contribute to the pharmacovigilance process, and what specific responsibilities do they hold? Thank you, Doctor.
Thank you, Bu Chairman, Bu Wisari Dila Samala. As I say before, many stakeholders are involved in pharmacovigilance activity, like health professional, government, industry, academician. And then also the patient and communities. Yeah. So in our context, as the researcher, we do research, and then give the must give the advice to the managerial, like the hospital managerial or health department managerial, dinas kesehatan managerial. So from there what we do, what we did, yeah. Become input for them to make the best policy, good policy in terms of patient safety. I think this is the most our contribute in our experience. All the study we did. Yeah. The hospital use this for minimum for the accreditation, because the in accreditation some indicator of education how hospital do they have the data, do the research and have the data related about the meso. So I think like this, probably Disari.
Amin. Okay, thank you so much, Dr. Dedi. Thank you, Miss Pepo.
Thank you, Dr. Sari and Dr. Dedi. Actually, there is one more question in the chat room. However, that we got limited time. So I hope that you can answer the question through the chat room, Doctor. So
Chat-
this I
chat
think
room ya?
will
Chat room.
yeah.
Chat
Yeah.
room, chat room.
Thank you, Doctor. So I think this is the end of our session for the session 3 in this move. Thank you, Dr. Dedi, for your informative presentation and thoughtful answer. And on
Okay, thank
behalf
you.
of the organizing committee, thank you, Doctor. We would like to present you with a certificate of appreciation for sharing your expertise with us today.
Thank
Let's
you.
present the certificate. All right, thank you so much, Dr.
Thank you.
Dedi.
Yeah, thank you. Nice for your as moderator.
Thank you so much. All right. Okay, ladies and gentlemen, this concludes session 3 of the ICCSCP 2024. We would like to extend our sincere gratitude to our speaker, Professor Dr. Pauline Lay and Dr. Apoteker Dedi Almasti for their valuable contribution. And thank you as well for all participants for your engagement and insightful question, which made this session both interactive and enriching. We hope you gain valuable insight from today's presentation, and we will look forward to seeing you in the upcoming session, inshallah. And thank you again for joining us. And I wish you a productive rest of the conference. And Assalamualaikum warahmatullahi wabarakatuh, and have a great day. Okay, I'll give back to the MC, Jihan.
Thank you very much, Apoteker Yoneta Srangenge MSc, for guiding us in this plenary session 3. And also, we would like to thank our amazing keynote speakers, Professor Pauline Lai, PhD, and Dr. Clean Farm Apoteker Dedi Almasdi. Ladies and gentlemen, as our appreciation for our moderator, we would like to invite Apoteker Yoneta to be on the screen to receive a certificate as the moderator in this third session of plenary lecture. Here's the certificate of appreciation and thank you to Apoteker Yoneta. Now,
Thank
please
you,
give
Jihan.
us your best smile, because our committee IT will take a screenshot for documentations. We start by counting from 3, 2, 1. Thank you. Once again, thank you to Apoteker Yoneta. Excellencies, distinguished guests, participants, ladies and gentlemen, we have now reached the next item on our agenda, which is Parallel Session 2. This parallel oral presentation will be subdivided into 7 separate rooms. To access each of these rooms, kindly navigate to the designated breakout rooms from the main session. Should you encounter any difficulties during the course of this conference, please do not hesitate to contact our committee through the WhatsApp group. As previously mentioned, we have a few breakout rooms within the pharmacy specialty, each featuring esteemed-invited speakers as indicated on the screen. Room 1 is Pharmaceutical Biology. Room 2A, 2B, and 2C is Pharmacology and Clinical Pharmacy. Room 3, Pharmaceutical Chemistry. Room 4A and 4B, Pharmaceutical Technology. Once again, to join any of this room, please access the respective breakout room from the main session. We are here to assist you, so feel free to reach out to the committee in the WhatsApp group if you encounter any challenges during this conference. We will see you again in this main room at 1 p.m. West Indonesian time for an upcoming session of plenary lecture. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Recording stopped.
Assalamualaikum warahmatullahi wabarakatuh. Welcome back to the second day of International Conference on Contemporary Science and Clinical Pharmacy, ICCSCP 2024, organized by the Faculty of Pharmacy, Universitas Andalas. Excellencies, distinguished guests, participants, Ladies and gentlemen, we are now continuing our conference to the next agenda. That is the fifth session of plenary lecture. This session will enrich our knowledge about the recent innovations in drug development supporting global public health by Profesor Apoteker Andi Dian Permana, PhD, from Faculty of Pharmacy, Universitas Hasanuddin, Indonesia, and Associate Professor Sukanya Dech Adisai, PhD, from Department of Pharmacognosy and Pharmaceutical Botany, Prince of Songkla University, Thailand. All right. First, we would like to inform that all audiences are invited to ask questions by writing the question on the chat room available, which the answer will be given during the Q&A session. For this session of plenary lecture, we will be led by Professor Renat Apoteker Dian Handayani, MS, PhD, as the moderator. Ladies and gentlemen, allow me to introduce you to our moderator. Professor Renat Apoteker Dian Handayani, MS, PhD, is an enthusiastic lecturer at the Faculty of Pharmacy, Universitas Andalas. She achieved her bachelor degree at Universitas Andalas and continued her doctoral degree at Julius Maximilian Universitat Würzburg. She has done some research and publication since 1997 in natural product chemistry and marine biotechnology. Her latest publications include fungal isolates from marine sponge, Chelonaplacilla sp, diversity, antimicrobial and cytotoxic activities in 2020, and isolation of endophytic fungi with antimicrobial activity from medicinal plant Rhodomyrtus tomentosa etenhas in 2023. Without any further ado, I would like to invite Professor Dian Handayani to be on the screen and guide the plenary lecture. To Professor Dian, the screen is yours.
Okay, thanks so much, Jihan. Assalamualaikum warahmatullahi wabarakatuh. Hello, everybody, everyone. Can you hear my voice?
Yes, Bu.
Okay. For this session, we have Professor Apoteker Andi Dian Permana, PhD. And before he start with his lecture, I will explain his curriculum vitae first. Okay, as here we can see, Professor Andi is a very successful lecturer in his career. I think he completed his undergraduate program in 2009 as the best graduate and completed his doctoral studies in 2019 in Queen's University, Belfast. And his latest research experience was conducted at the Department of Applied Chemistry, Faculty of Engineering, Kyushu University in Japan in 2031 last year. And he also underwent postdoc research at the Department of Biosciences, Chemical Biology and Biological Therapeutics at the University of Salzburg, Germany in 2029. And until now, Professor Andi has had 127 Scopus articles, amazing, with a total of 2,424 citations. And this afternoon, we thanks Prof. Andi for your sharing and short lecture on microneedle delivery system as a promising approach for improved systemic and local deliveries of drugs. You have 45 minutes for your presentation and continue with 5 minutes discussion. Okay, Prof. Andi, welcome. Time for
Hello.
your presentation. Hey, okay, Prof. Andi, how do you do? Nice to meet you.
Nice to meet you too. Well, we have
Okay.
actually the same name, Dian.
Yeah. How can I call you? Andi or also Dian?
Well, actually, everyone calls me Dian, so you can call
Okay.
me
Oh,
Dian
yeah.
also.
Okay, nice. Dian
Yeah.
is a good name, actually.
Yes, of course.
Okay, Prof. Andy. Prof. Dian,
Yeah.
your time is yours now.
Okay. Hello, everyone. Thanks very much for having me, and thanks very much, Professor Dian, for a really nice introduction. And let me share my screen. So, can everyone see my screen now?
Yes.
Okay. So, as I have a really limited time, only 45 minutes, so I will try my best to explain a brief, about several studies and several research about what I have done regarding microneedle delivery systems as a promising approach for improved systemic and local deliveries of several drugs. So yeah, I am Andi Dian Permana, and everyone calls me Dian. I'm from Faculty of Pharmacy, Department of Pharmaceutical Technology, Hasanuddin University. And before I start explaining about microneedle, so let's start talking about skin, because skin is one of the most important part of the body that can act as a drug delivery route. Why? Because as we know that the skin is the largest part of the body, it has a really really wide surface area. So when we apply the drug, so it's a non-invasive and really convenient route for drug administration. And also, compared to other drug delivery routes such as oral administration or injection, so skin is suitable for both systemic and localized drug effects, which I will explain why. And also, by delivering the drug to the skin, it can bypass first pass metabolism. As we know that oral administration have several problems in terms of metabolism that can decrease the effectiveness of the drug. So instead, when we use skin as a drug delivery route, we can bypass first pass metabolism. Hence, we can improve the effectiveness of the drug. We can reduce the elimination part of the drug, for example. So in several mechanisms. And also, by using skin as a drug delivery route, we can control the drug release, and we can reduce the dosing frequency, which can improve the convenience of the patient. And also, easy self-administration, because it doesn't need specific tools or specific health professional action like a physician, nurse, or pharmacy, for example. So everyone can use skin as a drug delivery route with easy self-administration. And however, several challenges in transdermal drug delivery system when we use skin as the drug delivery system. So we have stratum corneum barrier. So this barrier can prevent large molecules from passing through, because the physical characteristic of stratum corneum can prevent several drugs to permeate through skin. And also, it require several requirements for effective skin penetration. Like it can only permit several drugs which has several molecular weights, certain melting point, and cell sign lipophilicity. So several drugs that don't have those kind of physical characteristics. So that's why we need some tools to improve skin permeation. And also, using skin as a drug delivery route, it it is limited for certain drugs because certain drugs are lacking of these other properties. So that's why like not all the drugs can be used for transdermal delivery system. That's why we use microneedles, because when we use microneedles, we can we don't care about the physical characteristics of the drugs, because when you use this, we can help the drugs to permeate the skin. We can deliver the drugs to the blood circulation under the dermal layer of their skin. So what microneedles are? So it's a new platform for drug delivery system. So it consists of micro-sized needles with various varying diameters, length, and made from different types of polymers. So let's talk about several types of microneedle. Actually, when you are asking about microneedle, for example, can we imagine like a patch, like a simple patch? It has like a small needles on the bottom part of the patches, and then it can help the penetration of the drug by simply attach the patch to the skin. So it's like that. So actually, we have several types of microneedle, like solid microneedles, hollow microneedles, coated microneedles, dissolving microneedles, and hydrogel forming microneedles. So solid microneedles, it is used only for pre-treatment. So it is not only drug delivery proper drug delivery platform, because like this part like it only help creating the holes through the skin, and then after that we remove the microneedle and then we put the dosage form like a patch or cream. So maybe some of you guys already like I always go to the cosmetics shopping or something like that. They can derma roller, so we can use derma roller to improve the permeation of the serum of the gels to the our face skin. So it's part of the solid microneedles. Also, we have hollow microneedles. So this part can help the permeation of the drug by creating the holes, and then the liquid of the drug can permeate the skin through the small holes and then go to the dermal layer of the skin. And we have coated microneedles. So what's coated microneedles? It's like needles, and then the drugs are coated through the needles part, and then it can help permeating the drug. And dissolving microneedles. So this is the part that I will explain more, because I explore a lot several types of the drug using dissolving microneedles. So by looking at the name, dissolving microneedles, so it is like a needles that is dissolved after administration through the skin, because the needles are made by the polymer, like a biodegradable polymer and biocompatible polymer. So when we apply the needles through the skin, the needles will dissolve, and then the drug will be delivered to the dermal layer of the skin. And then after achieving the dermal layer of the skin, the drug can permeate to the blood circulation and then go to the part of the body. So it's simply like that. And we have hydrogel forming microneedles. So it is the needles that can absorb the interstitial fluid of the skin, and then it can swell. And then this part doesn't contain the drugs. So using hydrogel forming microneedles, we put the microneedles on the skin, and then we put the drug reservoir on the top of the skin. And then after the microneedle absorb the interstitial fluid, the drug can help. I mean, the drug can permeate through the swollen part of the needles. So it's like that. So it's not like dissolving microneedle. So what are the benefits of microneedles actually? So I will focus on dissolving microneedles at the moment. So it's actually painless and safe delivery, because even though they have needles, it doesn't reach the pain nerve under the skin. So it doesn't cause any pain, and it's really safe, because the hole created is really small, so it can it doesn't need any like a sterilization or uh, or 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 any related with the sterile dosage forms. And also because it is made by the polymer, especially by the degradable and biocompatible polymers, so it doesn't cause any sharp waste disposal. So compared to the needle itself, like injection needle, it's like a metal part, so it can produce sharp waste disposal. And also, the most important part is it's versatile for several types of formulation. So we can include several types of the drug inside the needles. So we can play with the composition, we can play with the ratio, and we can put any kind of the drugs inside the needles. And as I mentioned before, because it's safe delivery, because it can reduce the risk of irritation and inflammation. Because even though it is permeating through the skin, several studies have shown that it doesn't cause any irritation and it doesn't cause any inflammation of the skin. So, how we prepare this kind of systems or dissolving microneedles. So there are several methods to fabricate the needles. So we have a two-step casting methods, we have the droplet-borne air blowing methods, and then several types of that. So basically, it's really simple. But what I have done here in my lab. So we use two-step casting microneedle. So what we do is that we have the microneedle mold, and then we make the drug formulation by mixing the drug polymers and then dissolve in water and then mix the drug to the drug polymer and then mix well and then centrifuge to remove the air bubble because air bubble can have several problems in microneedle fabrication. So when we have drug formulations, we just pour the drug formulation at the top of the mold, and then we put the holder ring to prevent the spreadness of the formulation when we centrifuge the formulation. So we centrifuge and then we remove the ring and then we make it them dry for 1 to 2 days. So it's as simple as that. But the most challenging part of the fabrication is how to how to find the suitable formulations because we can easily produce the microneedle, but the challenging part is how to make the hard needle, how to make the like a really sharp needle, how to make sure that the drug can permeate the skin because it's the most important part of the microneedle formulation. So there are several types of evaluation of microneedles, but now I am focusing on mechanical and penetration properties because it is the most important part. This is the most important characteristics of microneedle because when we have the needles, we have to make sure that the needles can permeate the skin effectively by evaluating the mechanical and penetration properties. So simply we have needles and then we have a mechanical sensor or we have a texture analyzer here in our lab, and then we make a several pressure like a 32 Newton or around 30 to 40 Newton, and then we apply the needles and we evaluate how the needles can resist by the pressure. So we have to make sure that it can resist the pressure because the needle have to permeate the skin, and then the needle have to be able to deliver the drug inside the skin. For example, because in the application we cannot see whether the needle is inserted or no because when you apply the skin once we apply the microneedle on the top of the skin, that's it. We cannot be sure whether the needles can permeate or no because we cannot see after application. But by using this, by evaluating the mechanical properties, we can make sure that okay, the needle is strong enough to permeate the skin by resisting the pressure 32 Newton because several studies have shown that manual pressure by hand like this, so it's equal to 32 Newton. So when we apply the needle to the 32 Newton pressure, and then the needle can resist the pressure, we can make sure that the needle can permeate the skin effectively, thereby delivering the drug to the skin effectively. As simple as that, actually. We have several types of characterization, but because limit of the time, I will focus on several results of the study. So let's discuss about the use of dissolving microneedle for systemic and local deliveries of several drugs. So I will show several of my studies that show that this kind of needles can be used both for systemic and local deliveries. So the first study that I will talk about is the use of solid lipid nanoparticles, which is incorporated into dissolving microneedle for lymphatic targeting. So here, well, I'm not going to explain all the studies, but I'm just explaining about the main part of the study. So here we aimed to deliver the drug to the lymphatic system. So what we have done was that we first formulated the drug to the lipid nanoparticle because to fulfill the requirement of lymphatic targeting, which is the high lipophilicity, the small particle size. So that's why we chose solid lipid nanoparticle to encapsulate the drug inside the lipid, and then we incorporate the solid lipid nanoparticle inside the dissolving microneedle, and then we deliver the solid lipid nanoparticle into the skin layer, because in the skin we have not only blood circulation, sorry, but also we have lymphatic capillaries. So that's why, by modifying the type of nanoparticles, we can avoid the drug to be delivered to the blood circulation, and then we can deliver the drug to the lymphatic system, because the aim of the study is to improve the treatment of the lymphatic filariasis. So that's why, we deliver the drug to the lymphatic system. So here we have, let me show the pointer first. Okay, so here we have lyophilized solid lipid nanoparticle, and then we mix with the polymer, and then we formulate it into solid lipid dissolving microneedles, and then we have these three types of microneedle. Actually, we have three types of the drug. Uh, ABZ is albendazole, DEC is the ethyl carbamazine, DOX is doxycycline, because we combine three drugs for the treatment of lymphatic filariasis. And this is the pictures of the needles that we have. So we use polyphenol pyrrolidone and polyphenol alcohol for the formulation. So for those who wants to see the publications, you can see to this website, or this is the title of our publication. And so we have two main results here. So the first picture that you see here is the plasma concentration, which we didn't want to target. And the second one, we have lymphatic target, which what we wanted to target. So here you can see the dark the dark line is the oral administration, and the dashed line is the microneedle delivery systems. So we can see here that by using microneedle in combination with solid lipid nanoparticle, we can see less drug delivered by microneedle, but we have a lot of drugs in the plasma using oral administration. And then we didn't want to achieve this oral plasma, because the target is not in the plasma. Otherwise, when we looked at the lymphatic concentration, so you can see here the dash and the dark lines. So you can see that the microneedle can improve the delivery of the drug in the lymphatic system compared to oral administration. So here we can see that by delivering this, so we can reduce the concentration of the drug into the systemic circulation, which is the plasma, and then we can improve the concentration of the drug in the lymphatic system, which is the main target of this. So this is the first study that show that we can improve the systemic circulation of the drugs using dissolving microneedle. And then in this part, we combine with the solid lipid nanoparticle. And the second study that I will show here is we have a nanoparticle which had bacterially sensitive properties. So what mean here what we aimed is was that we would like to incorporate the drugs, which is doxycycline in this case, into the bacterially sensitive nanoparticle. So we encapsulate the drug using the polymer, which is sensitive with the presence of the bacterial, and then we can avoid the drug release when no infection. So it can reduce the exposure of the drug, which does of the drug to the healthy part of the skin, for example, in this case. So here we incorporate the nanoparticle inside the dissolving microneedle. So it is for the treatment of bacterial infection, especially for the bacterial biofilm. So here, in the first picture that you show in the screen is the formulation of the microneedle, because it is yellow because the drug is doxycycline. It has yellow color. And then it is the examples of the infection model that we have, the biofilm skin infection that we have in the here. And then you can see here in the part, the first part is here. So the first pictures is the normal skin, okay? And then the 2, 3, 4, and 5, these are the condition where the infection occurred. with different types of the bacterial. So we have Staphylococcus aureus, we have Pseudomonas aeruginosa, and then we have like the two types of Staphylococcus aureus and two types of the Pseudomonas aeruginosa. So we have several types of skin infection in this study. So the red here, the red line, how to say this, the dark red, because we have two red, dark red and light red. So the dark red is a microneedle containing free doxycycline. So doxycycline without any encapsulation inside the nanoparticle. And then we have 5 formulations of nanoparticle here. So you can see here the healthy skin without encapsulating. We can see like the drug is show the high concentration of the skin, while when we encapsulate inside this nanoparticle, when we encapsulate the drugs into the nanoparticles, in the healthy condition, no drug release. Like we have really, really small amount of the drug release, meaning that we can avoid the exposure of the drug to the healthy skin, to the healthy part of the body. Meanwhile, if we saw the pictures with different types of the bacterial, we can see that by the presence of the bacterial, by the presence of several types of bacterial, so we can see like a really, really high release, really, really high release of the drugs, meaning that the presence of the bacterial can stimulate the release of the drug because the polymer that we use is, which is polycaprolactam. Maybe you are wondering what's polymer we use. We use polycaprolactam, which is the polymer that can be affected by the presence of the bacteria. So that's why like we can see really, really high concentration of the drug here. And the third study that I'm going to show you guys is like the albendazole nanocrystal. So if you want to see this is the site of I will share the material actually, so you can see the titles and then you can search for the publication if you want to read more about the study. And the third study that I will show is the formulation of albendazole nanocrystal into dissolving microneedle for improved pharmacokinetic performance in the treatment of cystic echinococcus. So here we have first pictures are the microneedles, and then the second picture says the dermatokinetics, the concentration of the drug inside the skin. And then here, the most important part of the study is this part is the concentration in the plasma. So here we formulate the albendazole into the microneedle, and then because why nanocrystals? Because albendazole actually it has two problems. The first problem is the solubility. The second problem is the first fast metabolism. So we improve the solubility by formulating albendazole into nanocrystals, and we avoid the first pass metabolism by changing the formulation from oral administration to the dissolving microneedle administration. So here we can see here, like we have 3 lines. The blue lines is albendazole. The red line is the first metabolite of albendazole, and the green line is the third metabolite of albendazole. So here A and B is A and B are the oral administration. C and D are the microneedle administration. A is the core suspension, meaning that albendazole without nanocrystals. C also. B and D is the nanocrystal formulation. So we can see here that using oral administration, you can see that the blue line, which is albendazole, we didn't see after 1 hour, meaning that oral administration can change the albendazole through the metabolites. So that's why we found the red line here. It's really, really quick, meaning that only within 2 hours, albendazole is metabolized to albendazole sulfoxide, which we didn't want in this treatment. So that's why, like in this treatment, the patient should consume really, really high dose, which produce several adverse effect in this study. So meanwhile, C and D, we can see the macroneedle. So the blue line is the albendazole. We can see that we can maintain the concentration of albendazole inside in the systemic circulation after 72 hours, after 3 days. So this is one application, right? So here in this study, we only apply the microneedle on the first day, but we can detect the albendazole after 3 days of administration. So it's really promising study that we have at the moment. So that's why like we can preserve the concentration of the drug in the plasma concentration. Meanwhile, we can reduce the administration. You can see here we still have a really small concentration of the metabolites in the red line. So here the bioavailability we reach like almost 88,000% of the relative bioavailability dissolving microneedle compared to oral administration. So we can see like really high concentration of the albendazole compared to oral administration. This is one of the study that show that really effectiveness of the dissolving microneedle. Also, we have glucose response microparticle loading into a dissolving microneedle for selective delivery of metformin. So here we incorporate metformin, which is anti-diabetic drugs, into glucose response microparticle. So here we chose glucose response microparticle, meaning that the drug is released only based on the concentration of the glucose in the systemic circulation. So it is really useful for diabetic patients. So this is proof of concept study. So we can see that by using glucose response material, so we can improve the selectiveness of the drug by depending on the concentration of the glucose. So here we can see like we can maintain the blood glucose levels into the normal concentration by using dissolving microneedles. So here we have like a phenylboronic acid as a glucose response material to maintain the concentration of the metformin based on the glucose concentration. So now we have a collaboration with the company in the South Korea. So we use this type of approach for several drugs of diabetic oral diabetic drugs. So it's really attractive. Now we have sorry, we have dissolving microneedle mediated dermal delivery of arterial nanocrystals for cutaneous candidiasis. So here uh, we have a study aiming to solve the problem regarding cutaneous candidiasis infection, skin infection caused by Candida albicans. Here we similar to our previous study. So we formulated atroconazole into nanocrystal formulation, and then we incorporate nanocrystal formulation inside dissolving microneedle, and then we deliver the drug into the the dermal part of the skin, and then we used like a skin infection model in ex vivo models in the porcine skins, and then we can see like here you can see like the green line after 48 hours we didn't see any growth of Candida albicans. Meanwhile, using cream and then needle-free patch of isochronizal nanocrystal. So we can see like the fungi are still there. So meaning that compared to conventional dosage forms like a simple patch or conventional cream, we can improve the treatment of the cutaneous candidiasis. We can improve the concentration of the itraconazole inside the skin. Here we can see like in the 1, 2, and 3 pictures, we can see like we can observe we observe really high concentration of itraconazole inside the skins, because you know cutaneous candidiasis, the infection is inside the skins. It is not on the surface of the skin. So here sorry 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 the last study that I will show, maybe we have like a discussion later. So here we have enhancement inside specific delivery of carfacrol for potential treatment of infected wounds using infection responsive nanoparticles loaded into dissolving microneedles. Well, actually here um, similar to the previous study that I explained before. So here we use carfacrol, which is one of the natural product at the moment, like which has really high potential antibacterial activities. So we use carfacrol for microneedle formulations, and then we use specific delivery here also. So we use nanoparticle. We formulated nanoparticles using polymer that is specific for the bacterial release. So here we can see the needles, and then we can see here the pictures, and A and A and C. This is the pictures of using light microscope. This is the pictures of the needles using scanning electron microscope. So we can see like it is still in the sharp conical shape of the needles containing nanoparticles there. And then here we perform dermatokinetic studies to evaluate the concentration of the drug inside the skin. So here we can see that like the dark line, the solid line is carfacrol microneedles without nanoparticles, and then the dashed line is the carfacrol microneedles, but the carfacrols are incorporated into the PCL nanoparticles, polycaprolactone nanoparticles. And we can see here that by incorporating carfacrol inside the nanoparticles, so we can see that we can maintain the concentration of the drugs up to 24 hours. Meanwhile, when we use pure carfacrol without any microneedle formulation, we can see that it is really quickly eliminated from the skin. So meaning that because carfacrol itself really it has really suitable properties of the drug permeations. So we have to maintain the drug inside the skin by using polycaprolactone, because as we can see, like it has really permeation ability, really promising permeation ability, because if you have a look at several studies, carfacrol is also used by carfacrol is also used to enhance the permeation of the drug inside the skin. So because it has antibacterial properties, so in this study we use this to enhance the treatment of the skin infection. So not only that, so here in the figure C, we can see like the different concentration of the carfacrol inside the skin into three different parts. So we have the first bar is epidermis, the second bar is dermis, and the last part is the permeation. So here we have carfacrol microneedles, and then carfacrol's polycaprolactone nanoparticles with microneedles. So here we can see that by incorporating the nanoparticle inside, by incorporating carfacrol inside the nanoparticles, we can see that we can improve the concentration of the drug inside the dermis layer. Like meanwhile, when we use free carfacrol, so we can see that most of the drug are uh, permeating through the skin, which we don't want, because meaning that it is like it is in the systemic circulation, because it can bypass the skin easily. So we use nanoparticles to maintain the drug inside the skin. So here, well, actually, it's the end of the slide, but here, from several studies that we just saw, so we can see that the use of microneedle can improve the effectiveness of delivery of several drugs. So not only for systemic circulation, but also for local delivery. So, well, actually, when we want to formulate the drug into the microneedle, we have to like the first thing that we have to see is the physical and chemical characteristics of the drug. By investigating those kind of characteristics, we can determine what kind of polymer that we use for the microneedles, and also what kind of approach that we can choose, because we have several types of microneedles, but in this part I only explain about dissolving microneedles. But maybe I have been asked about what is the limitation of microneedle. I didn't show in the slide actually, but um, some of you might be wondering what are the limitation of that. The biggest limitation of microneedle is the because of the size is really small, and then the needle is really short, so it is suitable for the drug with low dose. For example, if you want to deliver the drug with the high dose, for example, it is still challenging. But now I am currently working on that. Like, we try to modify several types of the needle to improve the loading capacity of the needle, so that we can load not only small dose drugs but also a high dose drug inside the needles. So I think that's all from me. I hope this can give you some informations or new knowledge or new paradigm about the micro delivery systems, because to the best of my knowledge, only several studies in Indonesia, only several researchers in Indonesia are working on microdose. So if you guys are interested working on this, especially for you guys are working in the pharmaceutical technology, you can contact me. The email in the first slide. So you can send me an email, and then we can have some discussion, because currently I have several I have several research project going on, and I'm open for collaborations with this project. So if you are interested with this, you can contact me by email, and then we can have a discussion to execute how we can collaborate about microneedle, because it's really promising, because it is delivery system that's really new in Indonesia, so it's really promising, and I think this really good opportunity to collaborate with pharmaceutical technology researcher in Indonesia. So thank you very much. I hope we can have a discussion with this session, and I hope you are interested with this study, and then we can collaborate in the future. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Very interesting presentation, Prof. Dian. And yeah, this is an update knowledge for me, especially maybe by this all audience here in the online session in this seminar. And okay, we now in a question and answer session. And in the chat wall, we have 3 chat. I read shortly. And okay, this is a question from Ninyoman Yudianti. She write, thank you, Prof. Dian, for the presentation. This is truly outstanding research. I have few questions regarding the dissolving microneedles. First, could you share a video or step that's showing the fabrication or formation process of the needles? I am curious about it. Also, could you let us know what equipment is used in the process? And the second question, based on your estimation research, dissolving microneedles are often combined with nanotechnology-based formulation, like colloidal dispersion. Is it feasible to combine the needles with non-nanotechnology formulation? If so, would this still be effective in enhancing product formation? Okay, thank you for the attention and your answer. Okay, Prof. Dian, this 2 questions.
Yeah.
Important question, I think. Please
Okay, yeah. So it's really important
answer
question.
directly, I think. Okay.
Yeah. So, well, actually, um, so here in our lab, we have two types of equipment that we use for microbial preparation. So sorry because I couldn't show the video because I don't have the video at the moment, but let me try to help you to imagine to imagine how this works actually. So the most crucial part of the preparing the microneedle is the mold. So I have collaboration with the Australian company, so they provide the mold for me, and then we have collaboration
Mm.
for that. So we have, but um we have the mold, and then we pour the formulation on the top of the mold. So after that, the formulation have to be we try to share like a whiteboard. I think so. Oh, I think no whiteboard at the moment. It's fine. So yeah. So here in the slide, you can see that when we try to prepare the microneedles, so we have several part of that. So in the first picture, we can see that the molds. So here, when we have the drug formulation, we put the formulation on the top of the mold. And then, what we aim in this study, what we aim in this part is to fill the cavity of the mold so we can form the sharp of the needle. So to fill the cavity, we have two options. The first options that I have in my lab is the centrifugation. So we have a specific centrifuge. So we put the mold inside the centrifuge, and when we centrifuge the mold, the formulation can fill the cavity of the mold, and then when we peel the formulation after 2 days, it's dry and then it can form the needles. And the second machine that we have in our lab is the pressure chamber. So the pressure chamber is that pressure that can help the formulation to fill the cavity. So it is really useful to help the permeation of the drug, to help the filling of the formulation inside the cavity of the mold. So the mold is itself is the most crucial part. And then we put the formulation, and then to fill the cavity, in my lab, we have two equipment. The first equipment is the centrifuge, and the second equipment is the pressure chamber. Pressure chamber is like an autoclave for sterilization without any heat. So it's just like giving a high pressure inside the tank so that the formulation can fill the cavity of the mold. As simple as that. So it is how we prepare the microneedles. And then also, so based on my experience, so microneedles can be combined with the nanotechnology, but also we can use pure drug inside the microneedle, not only pure nanotechnology. So because if we don't have any specific targeting, so if we don't have any selective targeting, we just want to, for example, we have the drug which have no problem in terms of solubility. And then we want to deliver the drug through the skin to the systemic system, to the systemic circulation. So what we can do, just directly put the drug inside the microneedle because we don't want the specific delivery. So the nanotechnology is used for the specific delivery, for the selective delivery. But if we want to deliver the drug to the systemic circulation without any selective delivery, without any selective targeting, so we can easily put the drug inside the microneedle if the drug has no problem in terms of solubility. This is this is
Mm.
this is the concept that we have to bring in our mind before preparing the microneedles. So that's why I said that when we want to make the microneedle, we have to concern first about the physicochemical characteristic of the drug. Because if we have no problem in terms of solubility, so we can easily put the drug inside the microneedles, and then we can apply it to the skin. The drug can be easily delivered the drug to the can be easily delivered to the dermal part of the body. So I hope this answers your questions. Thank you.
Yeah. Okay. Thanks so much. Okay, Ni Nyoman Yudianti, I think your question already answered by Prof. Andi clearly and details. And now we go to Bapak Sumardi Saat. He have question here in the wall, the chat wall to Prof. Dian. How to level pain in the skin when microneedle application?
So, well, actually, like based on several studies, so we don't have any specific levels of the pain because it's like I was the volunteer at the time in the clinical study of microneedle, and then like it is I didn't feel any pain at all when we apply the microneedle in my
Oh,
arm. So
really?
yes,
Very
yeah. Yeah, so
nice, yeah.
we can
Uh-huh.
see like the pain that we feel is the pain from the pressure, not the pain from the needle. So because
Mm,
it's pressure like this, so we can see like a really, really low level, but we have no like a specific amount or specific scale of that. So that's why like I couldn't answer actually the scale of the pain, but it's really like it's really painless. So just like this, and then we remove, and then that's it. So it's really painless because, as mentioned, that the needle is really, really short, and then it doesn't reach the nerve pain on the under the dermis layer, so it's painless. So yeah, I cannot say any specific levels, but it's painless.
Okay, thanks. Yeah. And then the last question from Maharani to Prof. Dian. I want to ask, is there an effect of using microneedle in treatment? Is there any special treatment used on the skin? And is there an industry that has tried it in Indonesia for the product you have developed so far? Oh, Maharani will use so so fast for this system treatment, Prof. Dian. I
Okay.
think I agree also with this question.
So
We have to make the product as soon as possible, Prof. Dian.
yeah, so
Please.
well, actually, because microneedle is not well developed in Indonesia yet, because it's a new dosage from Indonesia. So in Indonesia the regulation is quite as a pharmacist we know that there are a lot of regulation in terms of preparing the drug and producing the drug in the industrial pharmacy. So at the moment, several countries, especially in UK and United States of America, so they use microneedle for cosmetics and also for vaccination. So several vaccination already use microneedle for that. But in Indonesia, we have not done any industrialization or translation in the industry. But there are at the moment there are two companies outside Indonesia. So they now we are working on industrialization, but not in Indonesia
Not yet.
industry. So I have collaboration with Australia and South Korea. Sorry. So
Okay.
they we now develop microneedle delivery system for diabetes, for pain, and for several treatment, and it's industrial. So we aim for commercialization, but not for market in Indonesia, but for outside Indonesia because of that. But so yeah. There are two industries at the moment using the research they have done, but not in Indonesia, unfortunately. But I hope that Indonesia industry can use this system to improve the effectiveness of several drugs. Yeah, thank you.
Okay, thank you. And we have still one question, and we have also still 5 minutes for this session. And in the chat, well, I see here from Ibu Nova Shafni. I think Ibu Nova Shafni, you can ask directly to Prof. Dian. Please ask ask direct to Prof. Dian.
Okay, thank you, Prof. That is interesting research. I've already heard about your name and also about your research. currently is very, give many influence to our, especially in pharmacists. So since you talk about the physical characteristic of the materials, so I'm curious, what is that? I'm from pharmaceutical biology actually, and also I'm thinking about the you talk about also about the diabetes. Is it possible for the people with the gangrene? Also one of the issue in the diabetes currently also. So if it is applicable also for the gangrene patients. Thank you, Prof.
Yeah. So if we talk about the physical characteristics of material that we want to develop microdosage forms, so the most important part that the material should have a strong mechanical properties. So they have, for example, polyphenol pyrrolidone. If we combine with the polyphenol alcohol, they can form the hydrogen bonding, and then it can improve the mechanical properties of the material if we mix them and then we dissolve them, mix them, and then make it them dry. So this is like the most important part. I mean, the most important characteristic that we have to consider is the mechanical properties. As long as the material have the mechanical properties, it is we can form this material into the microdose. This is the thing that we have to consider. And then after that we can play with the concentration, we can play with the ratio and everything. So this is like a that's why I say like macronil itself is not really complicated because it doesn't have a really specific requirement, because the most important part of the needle is how what we want to formulate inside the needles. But how to form the needles is not that difficult, because as long as they have a really strong mechanical properties, we can choose them as a material for microneedle. And talking about the gangrene diabetes, so one of the study that we have at the moment, even though it is just ex vivo study, is that gangrene diabetic infection. Because I have one study at the moment, we just finished, but it is like just ex vivo. We want to continue to the in vivo next year probably. Is that for diabetic foot infection? Because we want to deliver the drug inside the infection, and then without surgery or cutting the part of the skin of to make them something like a really really disgusting part of that. So we can use needles to deliver the antibiotics on the top of the skin, and then it can help the drug to be delivered inside the infection, and then the drug can work from the inside part of the skin without make them open, for example. So this is the study that we have at the moment. And then it is really interesting part of the study. And I think this is really versatile because skin is really rich with anything. It contains not only blood circulation but also lymphatic systems, and then we have also like immune cells. That's why like vaccination is really really crucial for delivering through the skin, because we have dendritic cells in the skin, we have Langerhans with the scales. So it is really versatile. So I hope that we can work with Nova, for example, so we can contact me and we can have a discussion after this how we can collaborate with the pharmaceutical biology. So yeah.
Okay. Yeah.
Okay, thank you, Prof. Dian.
Thank
I'm
you.
looking forward for your next research for sure.
Thank you.
Okay. Okay. Prof. Dian, due to limited time, we have to close this session. And thanks so much, Prof. Dian, for your interesting presentation, valuable lecturer that we have many valuable information for this session. Okay, before we close, I would like to summary your presentation. That microneedle systems involve arrays of tiny needles that penetrate the upper layer of the skin without reaching deeper tissues or causing significant pain. This needle can be coated with a drug, have drugs encapsulated within them, or be used to create microneck channels in the skin through which a drug is later applied. And primarily, it is used to transdermal delivery of vaccines, drugs, or cosmetics. The goal is often to improve the delivery efficiency, and patient compliance compared to the traditional hypodermic injection. Okay, thanks so much, Prof. Dian. I hope we can see again next time. And I will close this session, and this session I give back to the MC, to Jihan. Jihan, please, and thanks so much for this session, Prof. Dian and ladies and gentlemen, all audience. Assalamualaikum warahmatullahi wabarakatuh. Hello, Jihan. Okay, I think Jihan is not in this session, and Prof. Dian, please you can see before the certificate as the keynote speaker from our faculty, from our university. Thanks so much for your appreciation.
Dian, can you hear me?
Yes.
You still moderating for the next speaker. Actually, it's two speaker.
Oh yes. Okay. So I give the certificate of appreciation directly to you, Prof. Dian. Please, the organizing committee make a document for Pak Prof. Andi and his certificate. Click. Okay. Camera already?
Yes.
Okay, already. Take a picture from the certificate and the speaker. Thanks so much, Prof. Dian. See you again. Assalamualaikum.
Thank you.
Okay. Okay, ladies and gentlemen, we now open the keynote lecture 9. Here we have Associate Professor Dr. Sukanya Dets Adisai from Department of Pharmacognosy and Pharmaceutical Botany, Prince of Songkhla University, Thailand. Hello, good afternoon.
Good afternoon,
Ibu
Professor
Miss
Dian.
Sukanya.
Nice to
Hello.
meet you. Hello, did you hear
Yeah,
me?
nice to meet you again.
Yeah.
Do you still remember me?
Yes.
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 We have met for the first time in Aceh, right?
Yeah.
Okay. Actually, we are same as alumni of DIAD, right?
Yeah. Yes. And the
Okay.
second time for ICCSCP. Yeah.
Right.
Yeah.
Okay. 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 Before you start with your presentation, we will read your curriculum vitae for the audience. Dr. Sukanya finished her PhD in pharmaceutical biology in University Heidelberg in Germany in 2000. It is not 2005, right? 2005, right? Or before? This is fast. I'm sorry.
No problem. 2005. Yeah.
2005, yeah, actually the
Just finished.
2005.
Not finished yet.
Okay.
Next year.
Okay. Okay. And she have many publication. The new one is in vitro and vivo in silico analysis of molecular anti-pigmentation mechanism of selected thyrejuvenating remedy and bioactive metabolite. And another publication that she have published. And also as Prof. Sukanya also many experiences as invited speaker, especially, and also she is now as head of department of pharmacognosy in the Faculty of Pharmaceutical Sciences, Prince of Songkla University. Thanks so much, Sukanya, for your time to share about your experience, research result regarding the secondary metabolite for anti-diabetic type 2, right?
Yeah.
Okay, you
Thank
have
you, Professor Dian.
you have 45 minutes for give us lecture, a nice lecture for us, and then continue with 5 minutes question and answer. Okay.
Yeah. Can I share
Sukaja?
my
Yes.
slide?
Yeah. The time is
Okay.
yours now. Okay.
Yeah.
Please.
Okay. Moment. Could you see my slide?
Yes.
Okay.
But not okay. Yes.
Okay.
Okay. We can see now.
Okay.
Please.
So, first of all, I would like to thank the organizing committee for ICCSCP 2024 for inviting me to be a keynote speaker today. And so today my talk will be concerned about natural anti-diabetics from folk medicine to scientific research. So some is from the review article, some from is information from my research. So maybe interesting for somebody in this conference. And so my talk will have the content about, first I will give a talk for introduction of traditional remedies and herbal medicine example in Thailand, and also introduction about diabetics mellitus. And the second one, we will move to the anti-diabetic assay via the in vitro study for natural products. I will focusing on alpha glucosidase activity. And the last one will be the example of the traditional remedy or herbal medicine in Southeast Asia, also that Indonesia or some is also in Thailand. And so now I would give the information about herbal medicine in healthcare system in Thailand. In Thailand, normally that for the herbal medicine, first, the people in Thailand can use by themselves for primary health care. This is mean that the plant that they know the indication already, so they can take by themselves. In the other way, the government will take a list of the herbal medicine in the national list of essential medicine. For this book, normally this book is a medicine composed of the Western medicine and also herbal medicine for the hospital that the medical doctor can take a list for prescribe to the patients. And so from the list of herbal medicine product in national list of essential medicine, they will divide it into groups. The first group is the traditional preparation. Most of this group is a mixture of the herbal medicine, herbal plants, but the other way is come from the research and development. So it means that some plant that has the research or scientific support data, and they test everything already. So after that, they will make like a Western medicine. For example, they can do for a tablet or capsule. And so the people feel like that they use like a Western medicine. And Next, I will move to for introduction about diabetes. Why I interest for diabetes? Because as you know now, everywhere in the world, I think that we come to the aging society already, and most of the disease for the aging society is a non-communicable disease, or we call it is a NCD. That is normally is a chronic disease, and one of the disease is a diabetics. As we know that diabetics is come from the pancreas, does not produce enough insulin, or the body cannot effectively use insulin that it produced. And so insulin is very important because insulin can regulate the blood sugar. So if they have a problem for this, they can increase the blood sugar. It's called hyperglycemia. Most of the diabetics, normally we have two types, type 1 and type 2, but sometimes for the women, they can have the other type of diabetes. We call it gestational diabetes. That is some women they get when they are pregnant, but after give a birth, they is gone. But some they still have diabetes after give a birth. And so how we know that we have diabetics or not? Or we just only pre-diabetic patient. We not jump to the group of diabetics patient. We have to check our blood sugar. And normally when you go to the hospital, the doctor will ask you to check your blood sugar first. Maybe start with the fasting blood sugar. But sometimes you don't, for fasting blood sugar, you have to stop to eat or drink anything except water for at least 8 hours. So after you check, you will know that you can be, you are the group of normal people or pre-diabetics, or now you are diabetics. After we know that, we can make sure that we are diabetics or not. The medical doctor will ask you for hemoglobin A1C or the other test also. If you are lucky, you are a normal. But if you go to be a diabetic patient, you have to be careful. The doctor will take a look which kind of medicine that is good for you. Normally for the Western medicine, they have mechanism of action by decrease glucose production or decrease glucose absorption. Or the other way, they have mechanism for increase insulin secretion or increase peripheral glucose uptake. So all of the medical medicines for diabetics, they want to restore the normal glucose level in your blood. So some patients, some diabetic patients don't take only one medicine. For the regimen for diabetics patient, the doctor maybe take 1 or 2 or 3 medicine together. And from the research, how to go to the product. Normally, we start for the research by in vitro study, in vitro model. So it means that we work in the lab. For the in vitro model, we start with the enzyme-based assay. And the second one is cell-based assay. For enzyme-based assay and cell-based assay, we call it a wet lab. It means that you have to work in the laboratory. But the next one, you can work with molecular docking. This one, you work only with computer and software. This is good. No need to pay for chemicals. But anyway, we have to combine all the results from the in vitro model. And after that, we move to the in vivo model. This is costly because we have to work with animal. In Thailand, normally we use for diabetics, most of the animal that we use mice, rat, or something like that. And after that, if we get the good result, we will go to the clinical trial to the human before go to the medicine. So we have to test clinical trial for human, does it work or not? And how to work with natural product research. Usually we start with screening. If we focus for anti-diabetics, we also screening for bioactivity of the herbal medicine for anti-diabetics. And after that, we select the potential one to isolate or purification for the active compound. And after that, we take the active compound for test the activity once more. And after that, if the activity is good, we also correlate with the molecular docking also. If it's also good, we take the strong compound to work in the animal model. And after that, we will try with to make like a prototype formulation. So for example, you can make like a capsule or tablet and also go to clinical trial at the end to be medicine in the future. For anti-diabetic activity model, normally we start with in vitro also. In vitro that we start with the enzyme assay and also cell-based assay. And for my group, we start enzyme assay with alpha-glucosidase activity to look. What about alpha-glucosidase enzyme? Why we want to inhibit the activity of this enzyme? Because after we take a food, we eat or we drink or some, for example, we will have rice or bread on is a carbohydrate. Carbohydrate normally is a polysaccharide and they can increase the blood sugar after digestion. How to digest this is work with the enzyme. For example, in the mouth, we have amylase enzyme, but in the small intestine, we have alpha-glucosidase. If alpha-glucosidase is function, they can digest for polysaccharide to be a monosaccharide and to absorb to the blood vessel. And after that, if you measure the blood sugar, the blood sugar with a high level at the end after the meal, if we can stop the activity of this enzyme, it will give you the low blood sugar. Normally in the market, we have medicine in the market that available. The name of the generic name of this medicine is acarbose, or the trade name for the original one is glucobay from Bayer. In the lab, we use this one to be the positive standard to control the lab. When you want to determine of alpha-glucosidase activity, we use the colorimetric method. How to work? In the experiment, we start with the substrate. The substrate is para-nitrophenyl alpha-D-glucopalanoside. This structure like this, and after that we add the alpha-glucosidase enzyme. Normally, the normal reaction, this enzyme can digest hydrolysis, this one to be para-nitrophenol. That's provide the yellow color, and the other part is glucose. In the experiment, we want to determine the amount of para-nitrophenol. If we have the sample, or positive control, it means acarbose, and if the sample or acarbose is really active, can inhibit this enzyme, so the para-nitrophenol will be reduced. You can see the experiment is a pale yellow color or no color. That is mean very active to inhibit this enzyme. So there has one publication, one review article from one professor that reviewed the medicinal plant in Southeast Asia for anti-alpha-glucosidase activity that potential source in type 2 diabetes treatment in Southeast Asia. So some medicinal plants also that for my research also that I will show you. This is the data for the prevalence of diabetic in Southeast Asia. As you can see, Indonesia and Thailand, is quite similar, or we are close friend. So about more than 10%. The highest is come to Malaysia. Why? We have to look how to control it, not increasing the prevalence. I can show you the example of traditional remedy or herbal medicines for anti-diabetic treatment in Southeast Asia. Some plant, I think that from Indonesia, you also know that. The first one is finger root, or in Thailand, we call krachai. The scientific name is Bosendokia rotunda. The active compound for alpha-glucosidase inhibition is the name of D-O-methyl rotunda flavonocharcone. As you can see, the chemical structure here. In Thailand, for this plant, normally we use for cooking. And however, in the market that available, some is we will make like a tea, or some is like a drinking juice, and also they provide in the capsule also. The second one is moringa or drumstick tree, or moringa orenfera. This also in Thailand we use for cooking. And you can see from the picture, some they also fly with like an omelette with egg. Yeah, I'm not sure in Indonesia, maybe also you know that, and you also use for cooking. They have some report that they use ethanol or methanol from leaves extract from this plant that exhibit the alpha-glucosidase inhibition. And after that, they would like to find out the bioactive compounds in this plant. They found that the active compound is with also flavonoid, as you can see in melicidin, quercetin, and camphorol. All of this is flavonoid compounds. And they have the report that this plant is also good for antioxidant. They enhance intracellular antioxidant. If they are good for antioxidant, they also can help for reduce blood sugar, and also how to say, control also diabetic symptom. And the third one is gambia. Gambia is the name of the extract. It's a water extract from the leaves or branches from this plant, Uncalia gambia. In Thailand, we call it katichu. From the extract, we call katichu. And also the Thai people use it by chewing with areca and betel, and the leaves of betel, and for tanning or dyeing. Yeah. And number four is a bitter melon. This is really famous in Thailand for control blood sugar. Momodica chalentia, but it's not good for us because it's very bitter. Bitter taste also for young people, they don't like it. But okay, for the old people, a little bit it's okay because the bitter taste. In the traditional medicine, they use the fruit from this plant for anti-diabetics, anti-fever, and bitter tonic. And the leaves of this plant also used for anti-fever. But we have to say warning for this plant. If you use together with insulin or oral hypoglycemic agent, because for Thai people, normally when they go to the hospital, if they have diabetics, the medical doctor will give them the regimen for Western medicine to control blood sugar. And after that, they would like to use also together with the herbal medicine. But after that, when they use together, they have synergistic effect. Synergistic effect that show is mean that lower blood sugar for the normal, and they can get how to say, some they has a headache, some they has a seizure. Yeah, that is not good. Have to be careful. Have to check the blood level, the blood sugar level. Yeah. And also that we have to say that warning for the patient that has a liver disease. They have some reported that if you use bitter melon, you eat bitter melon together with a patient that has liver disease, they show hepatitis. Have to be careful. After that, some publications also report the chemical compound that show the effect for blood sugar level. For this bitter melon, they show that they has compound is chalanthine. Chalanthine is a mixture from cytosterol glucoside and stigmasterol glucoside. This is combined 1-to-1. The mixture have 50% of each compound together, and they show it's good effect for control blood sugar. But if they are they still in a glycone part, it means that they don't have a sugar part in the structure, no effect at all for control blood sugar. The second one that have in bitter melon we call is polypeptide P or P insulin. This is an unidentified insulin-like protein. This is similar to bovine insulin. The last one is a visin. Visin, this is an alkaloid glycoside. As you can see the structure here, this is have to be careful because it's toxic for the people that lost the enzyme GCPD. You know that some people they has a problem from this enzyme GCPD. They cannot eat some kind of peanut or something like that. This one also have to be careful when take this plant also. For the bitter compound of this plant is called that the cucurbitacin group. They have many compound in this group. They can reduce blood sugar also. And so other compound that is a report for this plant, for example, saponin polysaccharide. Many publication from this plant because it's a very good result for control blood sugar. So after that, many researchers onto interest with this plant. And this is overall for the mechanism of action of bitter melon for hypoglycemic effect. If you want to, if you interest from this, you can read more in the publication. And how to use this plant. You can use this by you take about 8 to 10 of this fruit and cut and take the seed out and cut in the small pieces, and after that you blend with about 10 milliliters from water and also drink 3 times a day. I would say that is bitter taste. And the food also you can make like powder, dry powder, and after that make like a tea also okay. But you have to be careful. Do not use in children and lactation women because it also can reduce blood sugar until it's seizure. So it's not good for two group of people, children and lactation women. Adverse effect, and that can happen for the patient that take a lot of bitter melon is a nausea and vomiting and numbness of the hand and feet. That is you have to be careful, and also hypoglycemic. It means that lower blood sugar. So we have to monitor also that when you use this, how much of blood sugar at the moment. Next one, pepper. Pepper in Thailand, normally for pepper, we compose in rejuvenating remedy. Also, that is the remedy for, how to say, for the old people, for long living also. In Thailand, we use the seed and also the leaves for reduce blood sugar. They has a report for animal model. But anyway, for this paper, I think from the publication, they have many benefit for human health. So why they also compose in rejuvenating remedy for long living, because they have more benefit. And when we're looking for the chemical constituent in this plant, we found that in the fruit, they had volatile oil. And in volatile oil, 1 to 2.5% composed with many chemicals. And in alkaloid, the major alkaloid is piperine. Piperine, as we know that this one is alkaloid. And why? The pepper is a spicy taste because they have piperetine. Piperetine that composed in pepper. That's why we get the spicy taste from that. Next one, Monbeli. Monbeli is normally we use the leaves, but for me, I love the fruit. When it's become black, it's really tasty. For the flu medicine, we use the leaves for antiperspirant or relief fever or relief heat. They make like a tea, as you can see this. But for the fruit, I think they have a lot of anthocyanin. This is also good for antioxidant. And for the chemical compound in leaves, we found flavonoid. Most of flavonoid is lutein, quercetin, maureen, camphorol. That's as I show for you here. But there has one that interesting. It's an alkaloid. The name is 1-deoxynolitrimycin or DNJ, or we call 1-DNJ. Some publication called duocustat or molanolin, the same of the name of this compound. They show the effect for alpha glucosidase inhibition is really good, and normally this compound is found in mulberry plants. But however, they also can found in also Bacillus species is for bacteria also. So this one is good from this plant to reduce blood sugar. And this is show the other compound in this plant. I go faster because some is not in time not enough. The number 7 is okra. Okra, or we call lady fingers. As you can see from the picture, the shape of this is like lady fingers. So it means that small and long nail. Yeah. In Thailand, normally we use a fruit of this plant because they has mucilate. They has pectin, they has scum in the fruit. How they use for treatment of stomach and also intestine disease. So for example, some patient has peptic ulcer. So for the flu medicine, recognize to use okra or lady fingers. In mucilate, if we look inside what is the active compound, they has acetylated acetic polysaccharide and galactolunic acid together. And after that, there has many publication to look inside this plant, the bioactive compound from different part of okra. They found that in the pot, they have polyphenolic compound, and in the seed, they also had polyphenolic compound, and the other root have flavonoid, leaves also flavonoid. But important for anti-diabetics, they found that the polysaccharide named as lamnogalactolunan. You can see the structure is very complex. This is active for anti-diabetics. And after that, they found more for not only the polysaccharide, they found also they has flavonol glucoside. This also active for anti-diabetics. Isoquercetin and quercetin 3-O beta glucopyranosyl glucoside. As you can see the structure as below, they found this in the seeds of okra. That is really active for alpha glucosidase inhibition. Number 8 is a hard leaf moon seed. If you see the leaf of this plant is the shape like a heart. Tinospora kisma is the scientific name of this plant. Normally, this plant is a climbing plant, and the part used is wine in the stem of this plant. Also, this plant is bitter taste also. Folk medicine use this plant for anti-fever, anti-diabetics, and bitter tonic. Because it's a bitter taste, so they use for bitter tonic. Normally also this plant, they compose in rejuvenating remedy also, because you can see that the pharmacological property that have been reported for this plan, many anti-inflammatory, immunomodulatory, cytotoxic, antioxidant, and blah, blah, blah. And important for us is also hypoglycemic effect. This is why they use also for anti-diabetic, but we have to say warning. We have to say warning, do not use this in the patient that has liver and kidney disease. For this plan, you have to be careful. For the chemical in this plan, many groups of chemical. For example, terpenoid, alkaloid, flavonoid, lignan, nucleoside, and sterol. In each group, what is the compound? For example, กลุ่ม terpenoid compound, they show the balacol, balapetol, balapetoside, and alkaloid, they have a berberine. But after that, they try to find out which compound that show the hypoglycemic effect. They found that bolapetol B and bolapetoside A and C in this. This is the effect for anti-diabetic to control the blood sugar in the patient. This is active compound. Number nine is liquorize, and liquorize, you can see that we can writing in two way for US style or UK style. Glycyrrhiza glabra, part used is a root, not leaves. We use the root of these plants in the folk medicine. Normally this plant we compose for mucolytic and soothe for the throat. In Thai medicine, Thai folk medicine, we compose in like a syrup when you have a sore throat or something like that. And for the Glycyrrhiza glabra or licorice, normally they are very sweet because they have sweetening agent. The name is glycyrrhizin, or you can call glycyrrhizic acid or glycyrrhizinic acid, the same compound. This compound is very sweet because there's when we compare with sugar, there's sweet more than sugar 50 to 100 times. So how come? You see, they take the licorice root, add in the food or herbal medicine or candy or sweet. You can, when you look, they say that they don't add sugar. You have to read that they add licorice or not, because some product is sweet because they add licorice instead of sugar. It contains at least 4 hydrophobic flavonoids, glabidine, glabon, and 4 palm or methyl glabidine. 4 of these compounds, they are effective for hypoglycemia, to control blood sugar. This is good for anti-diabetics. And also they have the report that glabin and glabidine is a phytoestrogen. Phytoestrogen is good for the women when we get the menopausal period. It means that you don't have the period at all. So in that time, is your hormone, estrogen hormone is dropped. So you need something from natural. Is phyto, is mean from the plant that can this compound can go to the same receptor with estrogen. So when you take this, it's similar to you get estrogen hormone. So it's good for them. And for licorice, you have not safe at all. We have to be careful also. For example, you have to be careful when you use it a patient that has hypotension or hypokalemia. Why you have to be careful? They say that do not use more than 50 grams per day over 6 weeks, because if you use a long time and too much, you will have a water accumulation, and after that your body will be edema because the sodium excretion is lower. It's lower, but the potassium excretion is higher. So this is make the patient increase the blood pressure. So why they say that don't use too much. And also this plant has a drug, herb drug interaction. When you use a diuretic, because of, for example, the patient that has a hypertension, when the medical doctor prescribe the medicine, they will provide the medicine for hypertension and also diuretic. So you have to be careful or to look that don't use together with liquorice, because when you use together with this, they can make potassium excretion is higher. When it's higher, it means that the blood pressure also higher. And avoid to use with diuretic, spironolactone, and amylolide, because they will reduce the efficacy of hypertension treatment. So I would say that not herbal medicine that is safe. It's the same like a Western medicine that you have to check also the patient. The warning for each patient different. It depend on each patient. The number 10 is Siamese cassia. In Thailand, we use also for cooking. This is come from my research also. Normally, flower and leaves we use for sedative. And the leaves and hardwood in the herbal medicine we use for anti-diabetics. In my research, we can isolate 5 compounds named as resveratrol, pisiathanol, dihydropisiathanol, chysophanol, and imodin. From 5 compounds, 3 of these have alpha-glucosidase inhibition, resveratrol, pisiathanol, and dihydropisiathanol. For chysophanol and imodin, they show the PTP1B inhibition. This enzyme also concerning for diabetics also. And resveratrol show DPP4 inhibition by molecular docking. It means that they do this work by computer. And on component against insulin resistance. So it means that says Senna wood extract on the component is good, has a potential for treat anti-diabetics. For resveratrol, if you look the chemical structure, the name of this group is stivine. Stivine is come from phytoalexin. It means the compound is can produce by several plant by response for injury or under attack for a pathogen. For example, that if they had bacteria or fungi, this plant will produce this compound. And also up to now, we can do by chemical synthesis also. Pisiathanol also, the group is still be the same. We found also the other fruit and vegetable. Resveratrol and pisiathanol is good for anti-diabetics and also is good for antioxidant. And for chysophanol and imodin, as you can see, the structure is an anthraquinone. Both of them is good for also laxative and also control the blood sugar. And we have to take a note a little bit for this plant, because normally part use leaf and budding flower we use for sedative and anxiolytic effect, and also to treat anorexia. But we have to be careful about chromoderivative, the balacol. This compound in this plant, they show the hepatotoxicity. In Thailand, after we know the balacol for sleeping, after that the people take Senna a lot. And after that, we have to take out from the market because the hepatotoxic. But if you eat for the food, for cooking, it's no problem because not high amount of balacol. And the last one is Mitragena speciosa, or we call it kratom. Kratom in Thailand, traditional use we use for headache, for pain, and also increase stamina, and also used for diabetics. many way for use. For example, you can make like chewing, smoking, or decoction. When you use it, you increase work productivity, make you more stronger, less sleep, less tired, more energy, and more active. And so you can use like a fresh or dry leaves and also effect in the same way. Most in Thailand, they use by decoction. And this is show the chemical compound in this plant, mitragynine and 7-hydroxymitragynine. This is the most important and it's an alkaloid group. And the problem for this plant, the chemical compound in this plant, because they can, they have the effect for CNS depressant, same as psilocybin or LSD and amphetamine. This is not good. And they have also the other compound is flavonoid compound. And the last, but time is enough a little bit. This is an example for publication concerning about traditional remedy for anti-diabetes for my research group. So at the end, I would say terima kasih. Thank you very much for your attention.
Thanks so much, Dr. Sukanya. How an extraordinary presentation that you delivered for us.
Yeah.
Amazing, amazing.
Hopefully that have the benefit for you.
Yeah, of course. I think we already know again about the health plans regarding the diabetic drugs as the traditional drug, and also you explain about the important thing about the chemical compound, the bioactive compound. This is I like so much. And as okay, we go now to the question and answer session. And in the chat wall, I have one question, but I think audience can ask the question directly to Dr. Sukanya, please. Oh yeah, I see here to Prof. Yufri Aldi raise his hand. Okay, Pak Yufri Aldi, please mention your question.
I could not hear.
Pak Al, Prof. Aldi, please unmute your speaker. Okay, yes.
Still not here.
We still not hear your voice, Prof. Yufri Aldi.
Professor Yufri, I could not hear you.
Yeah.
Baik, alhamdulillah ketemu dengan
Assalamualaikum.
Ya. Your research topic is still diabetic mellitus ya. We at UNAN, my research is immunomodulator. More specific research on increased immunity again, pandemic the cell at the current attacks to world COVID-19. We have worked to activity plan on activity of NK cell, CD4, CD8, antibody, and T cell. What you like think we can do here? The plan to well test a moringa leaf ya, daun kelor, pegagan embun, dan daun sungkai leaf. Sekaya.
Excuse me, I'm not sure that I get your question or not. You mean that you work with moringa? Excuse me?
Okay. Prof. Yufri Aldi
Yeah.
just mentioned that he also worked with some traditional plants.
Mm-hmm.
It is also with moringa, but he worked into immodulatory
Immune
immune
system.
system.
Uh-huh.
Yeah,
Yeah.
he mentioned about that. And your answer, Prof. Yufri Aldi, is your question is,
Yes.
what is your question for Dr. Sukanya?
I heard that you say that you work with immunomodulator and from moringa plants. And what is your question?
Yeah.
Or you just only want to share that you also work with this
Yeah.
plant?
We have working on the activity plan of land to activity and cell, CD4 and CD8.
Okay. Yeah. Yeah. He
What
mentioned
do
that.
you think we can do there? Apa kira-kira yang bisa kami kerjakan di sana nantinya kalau karena kita dah ada kerjasama ini.
Oh, okay.
Yeah.
He has you, Dr. Sukanya,
Yep.
because the collaboration.
Yeah.
What can he do in your laboratory later if he want to make collaboration research about these plants or another plants from Indonesia from our
You mean
place.
that my facility or my research work here? So in my research group, we focus on
Yes.
the anti-diabetes. We have the cell
Yes.
base and enzyme base. And now we will start with animal model by zebra fish. Because first before we do with the animal model with rat, and now we move because rat for us is costly, and now we move to the fish. Is we call it zebra fish, the small fish. Yeah, zebra fish.
Okay. Zebra fish.
Yeah, and we try to do that by looking for the insulin secreting from that model. And for zebra fish is good. It's a model because it's a small fish and take not so long time. But for animal, you can have you have to do so long time. Yeah, but for fish, just only after they fertilize. And after that, you treat after 74 hours and looking, I think it can finish in 2 weeks. Yeah, for we can do for toxicity for the plant extract, and also we can do for cosmetic also, for example, for whitening agent, and also to look for immune system also. Yeah, we try to exhibit this model because now we set the lab already, but the protocol is not exactly at the moment. Yeah, but for Professor Yufri, so you say that you have the plant extract already, but if you do for immune system and you want to do for the other, for example, not only anti-diabetics that we do also antimicrobial. We do also the cytotoxic for the cell line. Yeah, and also we do for the biomarker. It means that we isolate the compound. For example, from Senna, this we isolate by ourselves the Senna, the compound from Senna.
Senna.
Senna, this plant, because in Thailand we use the leaves and also the flower, the budding flower for cooking. Yeah, cooking like the food, but for the folk medicine they use a hardwood. Yeah, for anti-diabetics. Yeah, so after that, we take this for extract and isolate the chemical compound inside, and we found five of this compound, and after that we take the compound to examine the activity again. We found that the most effective is physiotenon. Physiotenon is really active, for alpha glucosidase inhibition. Yeah. So this is our facility that we can do. We do phytochemistry and also bioactivity.
Yeah.
Yeah.
Yeah. Thank you, Miss
Welcome.
Skanya. Yeah.
Yeah.
See you
Okay.
again.
Yeah. See you again. If you want to visit us, welcome.
Yeah.
Welcome.
Okay.
Welcome.
Yeah.
Yeah. Then the next question come from Dr. Wisari. Okay. Ibu Sari, please.
Okay.
For the time.
Thank you, Prof. Dian. Hello, Dr. Skanya. Long time
Hello.
ago. Nice to meet you too.
Yeah.
Thank
Nice to meet you.
Thank you for such an enlightening presentation. I have two questions for you. The
Yeah.
first question, the growing prevalence of diabetes mellitus poses significant challenges for healthcare systems, particularly in Southeast Asia. What are some specific advantages of using traditional medicinal plants such as bitter melon and ginseng in the management of diabetes mellitus compared to conventional pharmacological treatments? And the second one, traditional medicine has a rich history of using herbal remedies for various health issues, including diabetes. How do the active phytochemicals found in these medical plants, like flavonoids, alkaloids, and contribute to their anti-diabetic properties, and what scientific evidence support their efficacy in controlling hyperglycemia? Thank you, Dr. Scott.
Yeah, I will try to answer your question. If I don't get it, maybe you can repeat once more. For example, from the first one, you say that how to do for the herbal medicine for anti-diabetics in Thailand or in any area. For example, for bitter melon and ginseng. In Thailand, normally that when the patient getting high blood sugar, and the medical doctor say that you are the diabetic patient already, and this group normally in Thailand they will start with the Western medicine. Western medicine to control the blood sugar, and the problem in our country that the Thai patient try to use a herbal medicine or like a dietary supplement to control blood sugar. But when they use together, the blood sugar, that if you use only Western medicine, they go to the normal blood sugar. But when they combine with the herbal medicine, they go down to lower blood sugar. That is a problem. So I say to the patient that if you want to use together, you have to say to the medical doctor, in order to adjust the dose for the Western medicine, maybe you can take lower amount of Western medicine to combine with the Western medicine. For example, in our project, we would like to combine, like for example, that alpha glucosidase inhibition. We have positive control is acarbose. This is Western medicine.
Okay.
And we try to combine that. If we use Western acarbose together with our extract combination, how about the effect? For example, that if you use acarbose alone, you get this inhibition. If you combine with plant extract, how about the inhibition? We try to adjust that. Yeah. In Thailand, I would suggest that if you want to use herbal medicine, it's good for the patient with pre-diabetics. Pre-diabetics is mean that FBS more than 100. FBS, fasting blood sugar more than 100, but not more than 126. If the patient say to me that, oh, now my blood sugar is really high, for example, that they say that they are really high and they check fasting blood sugar more than 100, but lower than 126. This group we call is pre-diabetics. So if they have a high blood sugar already, but not jump to the group of diabetics, we can use a herbal medicine first to control the blood sugar. So after that, they cannot jump to the group of diabetics because you can see some herbal medicine is like a food we can use for cooking. So just only that we have the project we call that food as herbal medicine.
Okay.
It means that you can take the foods as herbal medicine. Yeah. It's not you don't eat medicine, but you eat food. But that food is benefit for your body. That's why we say like that. So I would suggest that if you want to use a herbal medicine, please use before you are diabetics. If you are diabetics already, you have to adjust the dose together between Western medicine and herbal medicine. Yeah. I'm sorry that the second one is what is it?
Okay, I repeat again.
Yeah.
Traditional medicine has a rich history of using herbal remedies for various health issues, including diabetes. How do the active phytochemicals found in these medical plants, like flavonoids and alkaloids, contribute to their anti-diabetic properties? And what scientific evidence support their efficacy in controlling hyperglycemia? Thank you, doctor.
You mean that how to know the bioactive compound
Yeah.
in that? It's a good question because only the some herbal medicine that the group of the researcher interest to look, not all of that. For example, that that I report I show you in the presentation. For example, this Moringa, they interest. So after that, they isolate the compound, and we know that. But some we don't know that. Just only if they interest, the group of the researcher is can. But if we want to know the easy way, just only screening the group of compound. Yeah. But it's not easy to know that because it takes time for isolate the chemical compound. Yeah. The fast way now, they use LCMS. They take the extract and after that they use LCMS. And if you are lucky and they have the library, they will know that the chemical component inside. Yeah. If you use a classical chromatography method, it take, I think, for master or PhD student from my group, take at least 2 years for one plant. Yeah.
Yeah.
Almost the same with my research in doctor program, Dr. Skanya.
Normally, if we have the library, that is good. For example, that some plant we use a CCMS, some plant we use LCMS. Yeah. But most of them, if you use if you work, you did a project with phytochemistry, you have to isolate.
Yeah, okay. Okay, thank you, Dr. Skanya.
Yeah,
Nice to
welcome.
meet you too.
Yeah.
Thank you, Prof. Dian.
Okay. Okay, Dr. Sukanya.
Yeah.
It seemed to be we need more time to discussion. I would like to have more time to discussion with you, but unfortunately, I actually the time is over until
Okay.
15 o'clock actually. But
You can come to here. We can talk more.
I don't know. But, can we extend the time for 10 minutes, Bu Sari?
Yes, sure, Prof.
Okay. We have another question, Dr. Sukanya, from Dr. Dedi Almasti. He write in the wall, Prof. Sukanya, how many products from your team already distribute to the market? The diabetic product.
In Thailand, it's
How
not
many?
easy.
How many
If you want to,
products?
if you want to make the, if you want to have a product that available in the market, because in Thailand you have to register in Thai FDA. That is not easy. Normally,
Okay.
for our research, most of the research stop in the lab or publication.
Yes.
Yeah.
Yes.
If you want to continue with the available in the market, you have to register for dietary supplement. Yeah. Or you have to register in the herbal medicine, not the normal medicine. Yeah. For example, that I have one project that we combine with Korean ginseng together with Thai herbal medicine to make like a tea. Yeah. And also for that, and then for that it's still like from the research and for just only publication, not available in the market. As I say that the how to say, the way that is difficult is Thai FDA.
Okay, this is
I'm not sure that Indonesia that has the same problem or not.
Okay, this is the last question, Dr. Sukanya.
Yeah.
Besides you have the research and the result published in the Scopus article, for example, did you write also a patent for the new novelty for the new research result? Do you have many patents for
You
that?
mean for the patent? For the patent, I don't do that. I do. I did only publications. For example, that today morning, I get the new one, the new publication. Yeah. And so for us in PSU, for the lecturer, you can do two ways. One, they interest for patent. The other one, they interest for publication. Or you can do together. Normally for the patent, the group of the research that interest for this is mean that they make like a formulation. Something like that. I try to register one for the patent one formulation, but not for diabetics. That is for anti-inflammation. The cream for anti-inflammation. Yeah, but still on the process for that.
Okay. Thank you. This is actually it's same also here in Indonesia that the researcher can publish the research result in Scopus.
Yeah. From the results, most of the result we try to, because it's how to say KPI. You have to commit for the research for the funding. That for this work, the output from this work. As you get the scholarship, what is the output that you have to send? Most of them they ask for the publications.
Genau. Ich bin darauf sehr einverstanden.
And they some now is more seriously. They say have to be Q1 or something like that. It's not easy. It's not easy.
I know that. Yeah. Okay. Ich fühle mich das Problem and yeah. Do you still speak Deutsch, Dr. Sukanya?
Ein bisschen.
Ein bisschen. Ein bisschen.
Oh, guten Tag.
Oh,
Guten Tag.
Sometimes we have to speak again in Deutsch, yeah.
Only
Dr.
ich liebe
Sukanya.
dich. Important.
Ich dich auch.
Ich
Okay,
auch.
Dr. Sukanya. Nice to meet and to discuss with you in very good time, very short time. But
Hope to see you in Thailand.
Yeah, I love that. Maybe next year I have to to visit you in Thailand, in Bangkok, for shopping and for learning.
No, in Songkhla, not shopping. In the
Songkhla.
Bangkok, in Songkhla, you can shopping also in PSU.
Oh yeah,
Maybe you
yeah,
take a
yeah,
group,
yeah. In
come
in
together.
Songkhla. Okay.
Yeah,
Hopefully, Dr. Sukanya.
hopefully. Yeah, sure,
Hopefully.
sure. You can
Okay.
come with Dr. Yufri and Wisali.
Uh-huh.
Yeah, because
Okay.
he want to know the facility here. That
Yes.
he want to collaboration.
Yes. Okay,
Yeah,
thanks so much. Thanks so much, Dr. Sukanya. And
welcome.
now, this time, we come the end the session. We all thank to you for your sharing, for your extraordinary lecture. And now we will give you the certificate from the faculty, the certificate of appreciation. This certificate is awarded to Associate Professor Dr. Sukarnatis Adisai as invited as keynote speaker. Thank you, vielen Dank, and
Thank you
please
so
the
much. Ik lever dik.
okay,
Yeah.
okay, yeah, see you again. I give back this time, this session to the organizing committee for the for closing session. Okay, see you again, Dr. Sukanya, and thank you for the audience for this session participation.
Thank you. So,
Okay,
bye-bye.
Assalamualaikum, bye-bye.
Yeah.
Thank you very much, Professor Dian Handayani, for guiding us in this session. And also, we would like to thank you to Professor Andi Dian Permana and Associate Professor Sukanya Adisai PhD for all the knowledge. Ladies and gentlemen, as our appreciation for our moderator, we would like to invite Professor Dian to be on the screen to receive a certificate as the moderator in this session of plenary lecture. Here's the certificate of appreciation. And thank you to Professor Dian. Now,
Thank you so much.
Now, please give us your best smile, Boo, because our committee IT will take a screenshot for documentation. Okay, I will help by counting from 3, 2, 1. Cheese. Okay, thank you. Once again, thank you to Professor Dian Handayani. Excellencies, distinguished guests, participants, ladies and gentlemen. As we draw near to the end of this event, I trust that each and one of us has found our time together both productive and inspiring. Ladies and gentlemen, our subsequent agenda entails the Best Presenters Awards and the closing ceremony. At this juncture, we are honored to introduce Apotheker Suryati, PhD, the Manager of Human Resource, General Affairs, and Technology Information of the Faculty of Pharmacy at Universitas Andalas, who will announce the 2024. Please join me in welcoming Apotheker Suryati, PhD, on the screen.
Okay, thank you, Ms. Gihan. Okay, Assalamualaikum warahmatullahi wabarakatuh, and good afternoon, everyone. It has been an incredible experience over these 2 days of ICCP 2024, with a few inspiring presentations, stimulating discussion, and impactful research. We are really grateful to all participants for their contribution and dedications. Now it's my honor to announce the best presenter and best participant for ICCSCP 2024. For each of our field has exceptional presenter, but 2 stood out in a category. So we kindly ask the best presenter to turn on the camera, as we love to see and celebrate this moment with you. Okay. I will announce for the best presenter in the field of biology pharmacy. For the first presenter, The name goes to Arifi Tanya for her presentation on the isolation of secondary metabolite from endophytic fungus Aspergillus niger A1 and its antibacterial activity. For Miss Arifi Tanya, please turn on your camera.
Congratulations, Arifi.
Congratulations for Miss Arifi.
Thank
And
you, Miss.
the best presenter for the biology pharmacy goes to Mr. Mainal Hurkan for his work on the anti-cancer potential of Garcinia coa leaf extract, cytotoxicity, cell cycle modulation, and apoptosis induction in MCF7 HERS2 cell line. Okay, congratulations, Mr. Mainal. Please turn on your camera. Okay, now we move to the next field. The field is pharmacology and clinical pharmacy. The best presenter goes to Mr. Halilur Rahman bin Muhammad Khan with the title Retrospective Study on Accidental and Deliberate Pharmaceutical Poisoning Among Adults in Malaysia. So congratulations for Mr. Halilur. And the next best presenter for the pharmacology and clinical pharmacy goes to Intan Nur Rahmadani with the title The Cost and Utility of ACT Chemotherapy in Breast Cancer Patient in Dr. M. Jamil General Hospital. Okay, congratulations for both presenters in the field of pharmacology and clinical pharmacy. And the next, we announce for the field of pharmaceutical chemistry. The best presenter goes to Ms. Desi Eka Putri with the title of research Exploring the Cytotoxic Selectivity of Taxus sumatrana Bark Extract for the breast cancer therapy. Congratulations, Ms. Desi, for your achievement. And the next, for the field pharmaceutical chemistry, goes to Mr. Puja Adi Priyatna with the title of Identification of LC-MSMS and Docking Analysis Topoisomerase 2 alpha inhibition from kratom leaf Mitragyna speosa as potential anti-cancer agent. Congratulations to Ms. Desi and Mr. Puja for your achievement. And next, we move to the field of pharmaceutical technology. The best presenter goes to Muhammad Nasrul Siregar with the project on increasing the solubility of multi-component crystal of ethylenoxib and piperine by liquid-assisted grinding method. And the next for the pharmaceutical technology goes to Ni Nyoman Yudianti Mendra with the title Nano Emulsion of Champaka Flower, Magnolia alba oil as antibacterial candidate, optimization, characterization, and thermodynamic stability testing. Okay, congratulations for the all best presenter. And please turn on your camera. And we can take a picture for the best presenter.
Sorry to say, unfortunately, my video is not working, so I only my voice.
That's okay, Mr. Halilur. Okay, congratulations for all the best presenter. And next, we also want to recognize an individual whose engagement and enthusiasm enrich the conference experience for everyone. And we want to recognize for the best participant of ICCSCP 2024. The best participant goes to Apoteker Hidayah Karuniawati S.Pharm MSc PhD. Please turn on your camera, Apoteker Hidayah Karuniawati, and congratulations and thank you for your active participation. with the title of Miss Hidayah Karnawati, also presenter for one of the presenter with the title Prevalence and Determinant Factor of Illegal Sale of Antibiotic in Shop and Grocery Store. So besides she is one of the presenters, she also very good in participation in our conference. Okay, thank you everyone for your good participation during this conference. Ladies and gentlemen, as we come to the close of the ICCP 2024, we reflect on the impactful discussions, new knowledge and connection that have emerged from this event. Over the past 2 days, we have had the privilege to witness outstanding research, build collaboration and explore future direction for science and healthcare. So on behalf of the ICCP Organizing Committee, I extend our heartfelt thanks to all presenter, participant, sponsor, and our dedicated volunteer for making this conference a success. So a special thanks goes to our keynote speakers and esteemed judges whose expertise guided this event. So last but not least, for committee member, thank you for your time, insight, and the energy you have invested to make this effort a success. So as we part, we encourage each of you take the insight, gain here back to your institutions, share uniform knowledge, and continue build upon the idea exchange. So let's carry on forward to the spirit of collaboration and innovation until we meet again in the next ICCSCP. Thank you very much. And see you on the next ICCSCP 2025. Okay. Assalamualaikum warahmatullahi wabarakatuh. Okay, I believe
Waalaikumsalam warahmatullahi wabarakatuh.
We extend our heartfelt gratitude to Apotheker Suryati, PhD, for her pivotal role in concluding this conference. Excellencies, distinguished guests, participants, ladies and gentlemen, as your Master of Ceremony, Jihan from the Faculty of Pharmacy at Universitas Andalas would like to express our sincere appreciation for your active participation over these 2 days. I also extend my apologies for any inadvertent errors that may have occurred. Thank you. Till we meet again in the next ICCSCP. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you so much to all.
Waalaikumsalam warahmatullahi. Congratulations, organizing committee. Success.
See you on next
Seminar.
2025. Bye.
Bye.
Bye, everyone.
Foto dulu kita ya Bapak Ibu.
Okay.
Please open your camera, please. Ayo, dede panitia buruan. Hah?
Fotonya di
Share
mana?
screen aja, cepetin aja share screen-nya.
Bu Suryati.
Ya Ibu saya.
Ada yang mungkin belum dilakukan setiap saat.
Apa itu?
Ada nggak disebar kuesioner pada akhir sesi?
Bu Ketua.
Oh.
Ada Bu Sari?
Bu Ketua.
Ada Bu.
Nah itu Ibu lupa juga ngingetin. Selalunya kalau dalam setiap event itu ada kuesioner supaya nanti kita bisa mengimprove seminar kita tahun depan itu lebih baik lagi.
Iya betul, ketinggalan.
Begitu Bu Suryati.
Pakai Google Form apa dia?
Itu aja lah kuesionernya untuk disebar ke mahasiswa.
Pakai Google Form aja nanti gimana?
Kemudian
Pokoknya pakai G-form sana Bu.
Iya pakai G-form.
Iya G-form
Jangan
kemudian.
Di foto ya Bapak Ibu. 1, 2, 3. Oke, next slide. 1, 2, 3. Next. 1, 2, 3.
Gigi Ami kelihatan nah?
Masih ada satu lagi. 1, 2, 3. Oke, terima kasih Bapak Ibu.
Offline-nya ada, offline fotonya ada tuh.
Ditunggu GoFood ya ke masing-masing alamat ya Bu Sari.
Pak Salman, Bu. Pak
Dian.
Salman.
Ayo Pak Salman, ditunggu GoFood-nya Pak Salman.
Ya,
Kiriman
silakan
GoFood.
datang ke kampus Bu Dian.
Oh, Oke,
Iya,
Assalamualaikum.
Foto sebenarnya, Lena. Foto di aula. Siapa yang masih tinggal? Panitia.
Terima kasih Bu Sari, Bapak Ibu semuanya.
Terima kasih Pak Ridwan, Bu Suryati, Prof. Dian.
Ya, selamat semuanya. Good job. Semoga tambah sukses.
Amin.
Tadi lupa tanyain Prof. Dian itu, Sari. Prof.
Ya, Bu.
Dian dari Unhas tadi
Ya,
tuh
ya.
masih muda banget, Nak.
Belum nikah lagi, Bu.
Hah? Single masih mungkin, Nak?
Oh, iya.
Serius? Udah profesor?
Itulah, Bu. Kenapa ya masih single?
Berapa umurnya tuh, Bu Sari?
Masih berapa ya? Si 30-an nggak salah, Bu.
Sagan Ibu tanya tadi, bara umurnya dek masih muda banget.
Iya, Ibu tanya tadi lah, Bu.
Mohon maaf Bapak Ibu, ini kita bergabung ini.
Oh, dengan siapa?
Apa-apa.
Dengan siapa? Prof. Dian tadi?
Iya.
Nggak ada.
Masih bergabung dengan umum ini Bapak Ibu.
Oh ya nggak apa-apa lah.
Ya,
Cuma
semuanya sudah
ngomongin
dewasa
kan,
kok.
supaya dosen-dosen muda itu jadi seperti Prof. Dian, Prof. Andi Dian tadi, betul-betul cetar gitu.
Iya, cetar.
Dia udah profesor, dia postdoc, dia melakukan postdoc, banyak riset
Dian.
kolaborasinya.
Prof. Adi itu dia patuh dengan senior-seniornya itu. Jadi kita ketemu dengan dia di Lombok waktu itu.
Oh iya.
Ya.
Oke.
Jadi dia disuruh cepat sekolah, memang cepat dia. Segera dia.
Kalau enggak kan berang Bu sama Bu Elina.
Ya.
Oke lah, azan asar. Makasih Bapak Ibu. Izin
Ya.
live. Assalamualaikum.
Terima kasih Bu Dian ya. Terima kasih Bu Sari.
Terima kasih Bapak Ibu.
Ibu, Assalamualaikum.
Wah, sekali. Saya kurang semangat, Cici. Ambil saja. Ya, apa kira? Lecet lagi.
Izin meeting ya Bapak Ibu.
Nah, oke, ya.